¿Puedo enchufar aquí?
Sí, claro.
Debería haber hasta un alargador. Que si no lo encuentras, creo que en el aula sobraba. Sí, por si acaso lo necesitáis para... Ya. No sé si
Take a quick look at the presentation is working as supposed to.
Yeah, sure. Ah, sí, sí. You have here the stick if you need.
Okay. Should be beneficial also. Thank you.
So, bueno. Okay. Thank you. Okay.
Sí, ya
I think you can just leave it open, and then Danny that is doing the operation, fine-tuning thing, can leave it open first.
Okay, should we?
Yeah.
I already had a quick look that it looks how it's supposed to, Mike.
Does it work? Probably. Where are you gonna?
I think I'm going to stay here because I need to skip.
So let's play.
Run in front of the screen, but besides.
Yes. Shoot it or shoot it?
Yeah, shoot it.
Please, yeah. By name. Yeah.
Okay, 3 o'clock sharp. Welcome to this afternoon session of thesis presentations of Embrc. It's my pleasure to introduce first the jury of this session, Professor Alba Ardura from Oviedo University, Professor Trinidad Perez, also from Oviedo University, and Professor Manu Soto from Basque University, all research partners of Embrc. I'm Luana Monteiro from Ghent University, Belgium, also the coordinator of this challenging and fantastic master program. Starting the session today is student Mariana Schutte, who is going to present about the immunostimulatory effects of marine bacteria and bacterial endotoxin in sea spray aerosols. Mariana has 15 minutes. When it's about 5 minutes left, I will make a sign for you. When it's about 2 to 1 minute left, I will make a sign that reminds them you can see who notice me, but success, the floor is yours.
Thank you very much for this introduction. So yeah, my name is Fiona and I present about the immunostimulatory effects of marine bacteria and their endotoxin in sea spray aerosols. And the thesis work was conducted at the Flanders Marine Institute together with Jolmong Lee and Martin de Reke. And I would like to start this presentation by asking you to take a look at this picture and remember, when have you been the last time at the ocean and how did that make you feel? Were you happy and relaxed? Did you maybe meet some friends? Did you go for a little beach run? There's actually a lot of studies over the past few years that show a better general health of people who live in coastal environments, which is indicated by less chronic diseases, less psychiatric problems, and so far the favorite explanations for this are reduced stress and more exercise and these mechanisms that I just mentioned. However, the ocean as a source of a diverse microbial exposure is often overlooked in that context, even though actually 70% of the oceanic biomass is comprised of microbes. And there are two hypotheses that suggest how these microbes can actually benefit our health. which is firstly the so-called old friends hypothesis, which says that especially in our childhood, we need this exposure to microbes to prime our immune system because we live in cleaner and cleaner environments. And secondly, the so-called biogenics hypothesis, which says that a low level and frequent exposure to bioactive compounds, such as microbes, but also pollen, spores, or toxins, benefit our health by inhibit or downregulate certain cell signaling pathways that are involved in inflammatory processes. And how can we get in touch with these bioactive compounds from the ocean? For example, by sea spray aerosols, which are small particles or droplets between 0.1 and 10 microliters in size that are created by wind and wave action, as seen, for example, in this little illustration. So you can imagine wave action is entrapping little bubbles of air, and when they burst at the surface, they transport these biogenics to us up to several hundred kilometers, also in coastal environments where we can breathe them in. And if we think about these hypotheses that I just mentioned, it's of particular interest to take a look at bacteria and their endotoxin in sea spray aerosols as immune stimulants. In this way, endotoxins, also called lipopolysaccharides, LPS, is a cell wall structure or feature in the outer cell wall of gram-negative bacteria. And the majority of the bacteria in the ocean is gram-negative. And endotoxin is known to be a very strong immune stimulant. And in the human body, this immune response is mediated by the so-called toll-like receptor pathway, in short TLR, and that can be seen as the first line of defense against invading pathogens. That might be all a little bit abstract. So how does it work? For example, you breathe something in, and there are microbes inside that trigger this toll-like receptor pathway. This pathway is triggered, and don't focus on all of these different arrows. I would rather like to draw your attention to toll-like receptor 4, which is the receptor responsible to endotoxin binding. Whereas the interplay of toll-like receptors 2 and 6 is responsible to recognize other microbial components, for example, from gram-positive bacteria. And that's why we took a look at these toll-like receptors in the study. And once these receptors at the cell surface are triggered, they start a downstream signaling cascade. And one very important factor further downstream, a protein, is called nuclear factor kappa B, in short NF-kappa B. And this one is of particular interest for us because it's very important by playing a key role in the mediation of further immune responses and defense mechanisms against pathogens or microbial components in general. And while it's known that high doses of airborne endotoxin exposure can have adverse effects like lung inflammation, sea spray aerosols actually provide a way for a frequent and low-level exposure to microbes as suggested to be beneficial by the old friends and biogenics hypothesis, because marine bacteria are known to have a lower virulence than their terrestrial counterparts. And there's also one study with mice that has shown when they were exposed to sea spray aerosols, there was an immune reaction, but not these adverse effects like lung inflammation. However, so far there was no study that has linked marine bacteria and their endotoxin in sea spray aerosols and the human immune response. And this way we made it the aim of our study to see if they have the potential to contribute to coastal health benefits. And the way we approached this aim was to sample sea spray aerosols, measure the bacteria and endotoxin concentration in the samples, and finally expose human cell lines to the samples, and then measure the induction of pro-inflammatory genes of the pathway that I just showed. About the materials and methods, so the sampling of sea spray aerosols was conducted using this device. It's a so-called Coriolis microbial air sampler, which has the advantage that it can take in very high amounts of air in a relatively short period of time that allowed us to take replicates of 60 minutes each. And the way it works is that we've got this air intake marked by the arrow. And due to the high air intake, the collection liquid in the cone underneath starts spinning very fast. And due to the centrifugal force, the particles, the aerosols, are trapped in the wall and trapped in the collection liquid. And this way we have them ready for further analysis. And our sampling strategy consisted of 4 days of sampling at the beach of Ostende in Belgium, shown in this picture, where we took 5 replicates. And we compared these field samples with samples generated in laboratory conditions with the so-called Marine Aerosol Reference Tank, in short MART. That's an abbreviation I'm going to use a lot in the following slides. And the MART is basically a big tank that was filled at the morning of each experiment with 120 liters of fresh seawater. And then we've got a pipe system in the tank that recirculates water, and a plunging sheet that is created, which is mimicking wave action. And this way we have sea spray aerosols generated in this tank where we can collect them. For further materials and methods, we use the so-called RFC assay to measure endotoxins, which uses RFC, recombinant factor C, that is a protein sensitive to endotoxin binding. Bacteria cells were counted with flow cytometry, And for the cell exposure experiments, we chose 3 different human reporter cell lines, which are cell lines that are specifically designed to measure the induction of the genes that I mentioned in this first big slide with the different pathways. So HEC blue are human embryonic kidney cells transfected with toll-like receptor 4. Toll-like receptor 2 and 6 was the second cell line. And then THP1 dual cells are monocytes to measure NF-kappa B induction. But I think it's going to become more clear when I get to the results. And yeah, the way we did these experiments was exposing the C-spray cells to the C-spray aerosol samples. 20 to 24 hours of incubation later, we could measure a color reaction and quantify our gene induction. As for the results, we collected in total 20 MART and 19 beach aerosol samples, so 20 in the lab and 19 in the field, and we could detect endotoxins in all of them. However, the highest concentrations on average were observed in MART samples. So we've got here the endotoxin concentration, endotoxin units per milliliter on the y-axis. And as you can see, the MART, so the lab samples, had on average a higher concentrations, whereas the variability was higher in the beach samples. And that's most likely due to different sizes collected of aerosols, because on the beach, the bigger aerosols were most likely to fall down before they have reached our sampling device, whereas in the MART, the sampling device was really next to this plunging sheet that was created. And the bigger the particles, the more likely they are to contain more bacterial material. And the higher variability in the beach samples was most likely due to natural variability, because during 1 hour of sampling, we had different wind action, different wave action, so different amounts of aerosols collected. And if we take a look at literature, there's actually just 1 study so far that has measured endotoxins in sea spray aerosols besides us. And in comparison to that, we found very high concentrations, but yeah, it's important to keep in mind this is just 1 study we compare it with, and they used completely different methods, so a different sampling device, and they also didn't sample as close to the source as we did, so they were a bit further away from the coast. Relating or taking a look at the bacterial concentration in the samples, we found a similar trend. So again, the ones in the MART, so in the tank that I showed, were higher than the bacterial concentration in the field samples. And correlating that to our endotoxin concentration, so here endotoxin units on the x-axis and on the y-axis the bacterial concentration in bacteria per milliliter, both log10 transformed, we found a significant correlation, very high, which showed us that with an increasing bacteria number, we also had an increasing endotoxin concentration in our samples. Taking a look at the gene induction, so after exposing our cell lines to our samples and 20 to 24 hours of incubation, we found a substantially higher induction from our samples than the blanks. And I would like to show that with the example of toll-like receptor 4, so all of our samples on the x-axis, and gene induction measured at an optical density of 405 nanometer on the y-axis. And I would like to guide you through this illustration by showing that the negative controls, we used ultra-pure water for that, gave a very low induction, as well as our blank samples, which was the collection liquid not used in the Coriolis sampler. Whereas in contrast, the positive controls, which was pure lipopolysaccharide from the bacterium E. coli, showed us that the assay worked. And we had a lot higher induction by MART and beach aerosol samples than our blanks. And taking a look, and also something that we observed was that the MART samples provoked a significantly higher response than the beach samples. And taking a look at the other 2 cell lines that we used, so toll-like receptor 2,6 and NF-kappa B, those genes also showed that the samples induced a way higher response than the blank controls. And again, MART, so the laboratory-generated samples, induced higher reaction than the beach samples. And at this point, we already had the main finding that sea spray aerosols can actually activate the human immune response. But remembering the aim of our study, we also wanted to link that to the endotoxin concentration in samples. And for that, we correlated the endotoxin concentration, here log10 transformed on the y-axis, on the x-axis, to the 3 different genes that we measured on the y-axis. And we found a significant correlation between every gene and the endotoxin concentration. The highest one, however, for toll-like receptor 4, which is, as we remember from the beginning, responsible for endotoxin binding, whereas toll-like receptor 2 and 6, which is responsible to bind to other microbial components, so not endotoxins, also showed the lowest correlation to endotoxins. And finally, taking a look at the gene induction in reporter cell lines, if we could correlate that. For this, I don't want to use any more correlation plots, but rather this illustration that I used in the beginning. We found a way higher correlation between the receptors, so toll-like receptor 2 and 6, and toll-like receptor 4 on the surface, and downstream NF-kappa B. than between the receptor genes that we measured. And this is also something that we were expecting because, as I said, the toll-like receptors, they respond to different agonists, but they are both able to trigger downstream the same response. And to see if we can actually predict the activation of this downstream response with the receptor genes, we did a linear model. However, here it's important to note that we don't have causal effects. It's just a correlation because we use different cell lines. However, our model showed us that toll-like receptor 4, so the one responding to endotoxin, was the main predictor. The interaction of both receptors was less important, and 2,6 was a non-significant predictor, which prompted us to the finding or reinforced our observation that endotoxins actually play a central role in the mediation of the human immune response to sea spray aerosols, but that we also have other microbial stimulants, so mostly gram-negative with endotoxins, but also other microbial stimulants. And thirdly, very important, we have a response beyond receptor activation. So the sea spray aerosols didn't only start the immune reaction in the human body, but apparently there's also something going on further downstream. But thinking about that, what does that actually tell us? So what are now the health implications of the measured endotoxin levels and gene inductions? Actually, we don't know about that. That needs to be investigated because there is a big gap of knowledge currently between endotoxin concentrations and gene inductions measured and actual health outcomes. But if we remember the aim of our study, that was also not something we wanted to show. Our aim was to link, or see if there's a link between marine bacteria and their endotoxin in sea spray aerosols and the human immune activation. And to our best knowledge, that's actually something we could show with this study for the very first time, that sea spray aerosols actually interact with the human innate immune system, and this way have the potential to contribute to the coastal health benefits. And for that, I would like to give a short outlook, what we recommend for future research and future approaches. So firstly, it's very important to take a closer look at the actual coastal exposure levels. So our study, one study taking place in the Mediterranean, we need more information about the actual exposure levels of endotoxins, and also where in the human lung do these particles end up. And for that, we can use devices like this that split aerosols by size fraction. Secondly, we need to know more about the dose-response relationship, so what kind of endotoxin or gene induction level triggers what kind of immune response. Thirdly, additional biomarkers and improved models are needed because, as I said, these cell lines that we use, they're great to isolate effects, but there are no real lung cells, so we should also use lung cells to validate these approaches and use transcriptomic analysis and better models. And if we, up to this point, still observe effects, we can validate that and connect that with epidemiological studies and see how that correlates to actual health benefits in coastal environments. And I know that must have been a lot of molecular biology right now, so I would like to end with 4 main take-home messages and my conclusions. So sea spray aerosols are actually able to activate the human innate immune response. And in this regard, bacteria and endotoxin are suggested as the main causative agents. However, they have the potential to contribute to coastal health benefits, but we need further studies to place these results in a broader context. And with that, I would like to end this presentation saying thank you to Yun Mong and Martin for their amazing support and counsel during this work. Irina and Max from the University of Antwerp for help with the cell exposure experiments. Jana for support on the final stretch of this thesis work, and family and friends, especially Estelle and Niels, for keeping company during long sampling hours on the beach and filling the mart, which is also some kind of exercise, getting 120 liters from the beach to the lab. And if you've got questions, my name is Biona, and you find my contact data, manuscript, and so on at this QR code. Thank you for your attention.
Thank you very much, Fiona, for this very interesting presentation. Now I'd like to invite you to ask questions. Start. Yeah. Thank you very much for the presentation. I thought it very, very interesting and sometimes a little scary. So see how that can trigger some immune responses in. And the thing is, sometimes triggering immune response can have an effect develop much response and you can have any different immunotary disease.
Do you mean?
I mean, if you stimulate your immune system,
yeah, Yeah.
Sometimes it can be a weapon in chemistry because you can develop an immune system.
Yeah.
Do you think that this can be a problem?
I don't think so at this low level and frequent or low level response. It's like, thank you for bringing that up because, for example, when we think about this old friends hypothesis, well, it's suggested that or observed that children who grew up on dairy farms with a lot of endotoxin exposure or higher endotoxin exposure, they benefit from that by showing less allergies at higher ages. For adults, that same exposure is actually still controversial. But I think when we look at the ocean, it's actually in this normal setting, so not talking about harmful algae blooms, but really just that frequent low-level exposure. I think this is not a level of concern because, as I said in the beginning, marine bacteria, they are known to have a lower virulence. So there's actually some marine bacteria that were already used or that are in trials for drug treatments because they can actually inhibit cell signaling systems. My supervisor is right now in Belgium in a laboratory with these samples doing these inhibitory experiments. And I think there are more of the positive sides than that we actually have a risk if we don't have harmful algae blooms.
Maybe I missed it, but how are you sure that all the side effects from SSA something that is there?
That's also a good question, and that's something we wanted to rule out by really sample directly at the source. So with the MART, so that tank that I showed in the beginning, we really so in this way, we also took a sample of the laboratory background air because we were also concerned that air intake, 300 liters per minute, that tank was not sealed airtightly. So we thought, what if there is something in the air of the laboratory that influences the samples? But therefore, we always took in the beginning, while we filled the MART, a sample of the background air. And we actually found very, very low concentrations in these samples. So we could rule out that there is something else going on in the background air. And at the beach, we just sampled so close to the source, so really that you could sometimes feel the sea spray in your face, that we really knew, okay, this should be from the marine bacteria actually and the sea spray.
First of all, congratulations for the talk and the work that you did. Just a couple of curiosities, because it's not my field of expertise, but I suppose that the bacteria populations, they change depending on the health status of the environment in which they are.
Yeah.
So you selected one clean place, so that it's almost polluted place.
So I would say
the beach or something.
I would say about pollution. So the way we wanted to make sure or to relate these data about the bacteria, my supervisor was more than 1 year, or for the duration of more than 1 year, analyzing the bacterial community in the North Sea. And yeah, she found that actually the majority there is gram-negative bacteria, mostly alpha and gamma proteobacteria. So that's how we kind of know this is mostly the gram-negative bacteria from the ocean, the endotoxin that we got from.
My question goes, you compare
Yeah.
what you use for this approach, this tool, to improve the sensibility or ability of the immune system to be enhanced. You compare bacteria coming from a polluted place and coming from a clean place, what could be the difference?
I think that's very important to investigate. As I said, that different bacteria can have different virulence. So that's probably a next step to see what kind of bacteria triggers what kind of immune response. But yeah, that's something for then, I guess, follow-up research, because at this point we just wanted to see if there is this immune reaction. And now, in general, when you say, is it harmful, is it not harmful, that really needs to be investigated with the next steps that I showed, because so far there's not really research at all being done what kind of level causes what kind of reaction, and it's really difficult even to say what kind of marine bacterium has what kind of virulence, and if that answers your question, I hope.
Thank you.
Okay.
Thank you, Luana. First of all, congrats, Fiona. You are here and finished. Yes, that's all. But I have some questions because I think it's a very interesting work. Something new, something that maybe I could see, okay, it's a good idea, but I didn't have this idea, no? So
Me neither.
It's a very good work. I have one question about the lab aerosols with mass.
Mhm.
Why do you put this? What do you expect? Is water from the seawater the same seawater when you are doing the sample? No?
Yeah, exactly.
So what do you expect? What difference do you expect for this way? What can happen? Excess, right?
I think it was mostly interesting because there was, as you say, so little research done in that field to see, do these approaches differ? What can we maybe use best also for further studies? And yeah, for the field samples in general, it might be an idea, okay, it's always best to sample in the field because that's what people are exposed to. But at the same time, there's that big natural variability. So we had some samples with very low endotoxin concentration, some samples with higher. And so it was very interesting to compare that to the MART samples. And we saw that, for example, if we had 1 week, some higher endotoxin concentrations in the field samples, that was also corresponding to the MART samples. So it was for us very interesting and important to validate that we can use both approaches.
So do you expect that the bacteria are in the water, not in the air?
Yeah, that's the main point, that they are in the water. And we also thought about sampling. Oh, why don't we use water samples in the beginning? But there are actually a few studies that show that the bacterial community from the seawater differs from that one that's in the air. So they know, okay, these are the bacteria in the air. This is the community in the water. This is the community in the air. But it differs. It changes.
So combining between them, you can have the differences.
Yeah.
Oh?
Yeah.
Okay. How do you check the gene induction?
Okay, no, sorry. No,
The
no. I just need to get back to your question. So it's not that you mean that we can compare the bacteria community by using MART and beach. So the
Yes.
tank. No, that's so we expect the same community because we both sample the aerosols. So it's still that we want to have the same bacterial community. Yeah.
Okay.
Sorry,
Okay.
I just wanted to clarify that.
Don't worry.
Okay.
So the other question, how do you check the gene induction?
How do we check?
We're talking about the different
Yeah.
the aerosols can how do you check?
So that's maybe I go back to this pathway in the beginning. Might take a second, but so the way these reporter cell lines work is that they have really only the there we are. So these reporter cell lines that we used, so HEC-Blue, human embryonic kidney cells, they are designed and really just express or with this reporter gene construct. And so they start a signaling process, but there's one protein which is called SEAP, secreted embryonic alkaline phosphatase. And yeah, that's activated, but only if this receptor is triggered because that cell doesn't have any other receptors. And that works kind of similar with toll-like receptor 2 and 6, and also with the third cell line that we used. So in general, it's not directly that gene that is shown here, so not NF-kappa B that we measure, but another like protein further downstream, but we know it's that one activated because of the design of the cell.
Okay. That's all.
Yeah. Okay.
Very much. We have had questions from audience. Questions? Yes. Okay.
This one? Yeah. So NF-kappa B is very important because it has that potential to migrate from the cytosol in the nucleus, and this way trigger further responses. And as you can see here, I hope it's readable. Maybe I can even go two back. It says pro-inflammatory cytokines. And if we think about a human immune response, these cytokines are really the key element to mediate a further immune response. So they are responsible to up and down regulate other genes, get immune cells in certain places to tackle these microbial pathogens or in general microbes. So yeah, that's really a key component for further responses.
Any other questions from the audience? Okay, it might be a weak question, but talk about we did have a huge impact for us just going around the world, like for our human and our system.
You mean that there was a higher concentration that we measured compared to that?
Kind of goes on the
Yeah.
rest of it's not scary, but just think that walking and like direct reaction.
If it's too much exposure. Yeah. So for that, it's important. So yes, we measured higher endotoxin levels, but that other study used a different approach and also something that we don't know. So the endotoxins that we measured were in the liquid sample, and it's something else if you measure them. You have them collected in the liquid sample. Does that really correspond to the amount that we have in the air? And for that, there are other analysis to measure sodium, because if we think about seawater, we know the amount of sodium we have in the seawater. And if we measure the amount of sodium that we have in our sample, we can see how many aerosols have we actually collected. And so that's also something that would help us to harmonize for the amount that we have collected, and then see, is that actually corresponding to the amount of endotoxins that we breathe in?
We have questions. Students, we have maybe time for one little question. Okay. Maybe I can ask you, well, I had a question, but first was just wondering, would you think it would make sense in the future when the method is more refined to assess difference of exposures of different agents? Because, for instance, surfers are more exposed to the sea spray aerosols than recreational swimmers that are more in the shallow zone.
Yeah.
Have any thoughts about that?
That's a very interesting thought. So we were thinking, if we observe actually an effect, would it make sense to expose mice to sea spray aerosols, human studies or studies with human participants? But at that point, we already have these health data. If we think about health surveys, so this study took place in Belgium, and there are studies with school children assessing some key parameters about their health. So we could now, for example, see how did that change throughout the years, because these studies or surveys go back for quite a while. And this way we can also compare coastal populations, maybe with populations further inland, with drug sales. So there are studies that show, for example, in Brussels, that you can use the sale of respiratory drugs like asthma treatments as an indicator for pollution. And this way we could, for example, take a look at the sales or sale numbers in pharmacies in communities directly at the coast, further inland, if we see there some kind of correlations.
Definitely. I was more thinking on the exposure of agents that have far less exposure, like the overexposure impact.
Do you mean ages? So like the
human example, because it was an example, a surfer frequently more exposed to more intense sea spray aerosols than swimmers in a more shallower zone.
That's also a good idea. But there we would need to see that if you go swimming and surfing, you're not only in touch with what you breathe in, but in touch with the seawater. And that's a complete other story again as well, because the seawater colonizes, there are studies showing that seawater bacteria colonizes your skin for a certain amount of time, and that changes your microbiome in a completely different way. So I think if we keep to aerosol research, we should maybe keep the swimmers and people in the water out for the beginning, and first look at those walking on the beach and living in the coastal environments. But yeah, thank you for that thought.
Thank you very, very much, Liana.
Thank you.
Now, I'd like to invite our second speaker of this session. We're going to wait a little bit until the presentation is ready. More already.
Where are you gonna be, Greta? What are you gonna be? Are you gonna be right here? Are you moving there?
Maybe just here.
Maybe here?
Yeah.
Right. So it's my pleasure to introduce Xinqing Lin, who's going to tell us about microplastics transfer from the ocean to the atmosphere through aerosolization in the presence of bacteria. Xing, the floor is yours.
Thank you. Good afternoon, everyone. My name is Xinqing, and I'm here to present my work on microplastics transfer from the ocean to the atmosphere through the aerosolizations, specifically in the presence of bacteria. My research has been conducted at BLIS, the Fender Marine Institute in Belgium, under the guidance of my supervisor, Anna and Martin, and my mentor, Yun Meng. Before I start my presentation, just note that my thesis manuscript will be submitted in August, so the results are not yet finalized. So, as we are all aware, plastics pollution is one of the biggest environmental concerns in the world, and the larger plastic debris can be degraded into smaller particles, which can be separate into two categories based on their size. The microplastics ranging from 0.001 to 5 micrometers, and the nanoplastic ranging from 1 to 1,000 nanometers. This figure just shows the potential transport pathway of microplastics in the ocean through the surface drifting, vertical mixings, beaching, and settling. Microplastics have also been found widely distributed in beach, river, seawater, sediments, and even in the polar regions and the atmospheric compartment. However, until now, only the land-based source were considered for the resource of the atmospheric compartment's microplastics. So the recent studies shows that the microplastics can be transferred from the ocean to the atmosphere through aerosolization process, such as the bubble burst ejections and wave actions, specifically in the bubble burst ejections, which is shown in the upper figure here. So the sea spray aerosol, which are produced by the wave-wind interactions and can be transported into the atmosphere through the convective updraft. So if you look at the figures here, it shows that the breaking wave can cause the bubbles with the trapping air to rise to the sea surface layer, and the nano-sized particles are sprayed from the sea to the atmosphere through the bubble bursting. So these primary droplets will be produced by the bubble film burstings, and the water jet will form through the this process, and when the cavity of the bubble collapses. And the void left by the bubbles is filled with the water, and the water collision process will form the secondary ejections and the larger micro-sized droplets. And some of the micro and nano-sized droplets will return to the sea immediately. However, the other will still suspend in the air and can be transported by air during the aerosolization process. So the preliminary results from the paper published this year by my supervisor and the previous master students indicate that the MMP particles in the size range from 0.5 to 10 micrometers can be aerosolized by the bubble bursting. However, the role of microbiota is still not understood well in the surface layer of seawater. Therefore, to add to the current knowledge, the goal of our study is to assess if the presence of bacteria impacts the aerosolization of MMP particles. So, to test the aerosolization of microplastics in the presence of bacteria and microalgae, we decided to run the trial experiment first with this simplified setup. Since this is faster to show provide an overview and also to prove the concept. And also this will avoid the waste of time and resources. So we will use these fluorescent polystyrene beads with a size of 5 micrometers in diameter, since this is standardized with a simple shape to avoid the confounding effect of morphology, and also it is neutrally buoyant since it has a similar density with seawater. And we will fill this Erlenmeyer flask with 0.5 liter of filtered artificial seawater. And this setup is connected to the filter holder with the 0.1 micrometer pore size PTFE membrane filter, and further connect to this air pump for 5 hours with the flow rate of 4 to 5 liters per minute. So for the trial experiments, we have 3 treatments. The first one is an MPs only, and the second one is an MPs with bacteria using the Idiomarina lohensis, and the MPs with microalgae using Rhodomonas salina. And for all treatments, we have the initial density of 1,000 cells or particles per milliliters. And after the experiments, all the samples will be manually counted under the fluorescence microscope using the blue light filter. So the result of the trial experiments indicate that the number of particles aerosolized has the most in the MPs only treatments, followed by the MPs with bacteria and MPs with microalgae. So during the trial experiments, we also found some microsphere aggregation in the presence of bacteria, and this is from the seawater samples. So this figure shows the comparison of aerosolized microspheres across 3 treatments. The x-axis was the 3 treatments, and the y-axis is the number of aerosolized particles. And in the presence of bacteria, we have found that the result of the 97% decrease, and the presence of microalgae results in the 87% decrease. However, the results from the trial experiments were not accurate enough. That's why we further ran the experiments with this device called Miniature Marine Aerosol Reference Tank Minimart systems. And this system is more controlled and precise with the water wheel 20 centimeter diameter and 8 centimeter wide. And this will produce the plunging water jet to fall approximately 10 centimeter from the water wheel to the water surface. And this will simulate the natural bubble plumps, surface foam formations, and also aerosol producing mechanism active during the oceanic wave breaking. This is just a short video to show you how the Minimart works usually. So for the aerosolization experiments, each sampling we have 3 replicates here, and also 1 blank samples. So we also use a fluorescent strength beads, and we will fill the Minimart tank with 6.5 liter of filtered artificial seawater. The extra 0.5 liter is reserved for the water sampling. And this device is connected to the 4 filter holder with 0.1 micrometers pore size PTFE membrane filter, and further connect to the air cube for 24 hours with the flow rate of 30 liters per minute. And in the aerosolization experiments, we only have 2 treatments. The first one is the MNP only with 4 different sizes of microbeads, the 0.5, 1, 5, and 10 micrometers, and also the NMPs with bacteria using the Pseudo-Autoromonas species. And also the initial density for all treatments, also 1,000 cells or particles per milliliters, and also all samples were counted manually under the fluorescent microscope using the blue light filter. So this figure, it just illustrates different size of aerosolized microspheres from 0.5, 1, 5 to 10 micrometers. And all pictures were taken under the fluorescent microscope using the 10 times objective, except for the 0.5 micrometers. Since they are too small, so we use a 20 times objectives. So in the aerosolization experiments, we also found some aggregation of the microspheres in the seawater samples like this. And this figure shows the linear regression model of the aerosolized microspheres by the log transform particles. The x-axis is the log transform microsphere diameters, and the bracket is the actual diameters. And the y-axis is the number of the aerosolized microspheres. So in this figure, we separate 2 treatments into 2 different colors. The blue line indicates the MMP only treatments, and the orange line indicates the MMP with bacteria. So these 3 dots on the plot indicates 3 treatments for each different test size, and no samples were found in the blank samples. So this figure, we can observe that there is the negative correlation relationship between 2 variables for both treatments, and the high R-square value and the low p-value indicate the statistically significant negative relationship between the diameters and the number of aerosolized particles, which means when the number of aerosolized particles will decrease when the particle size increase. And also we observe the number of particles aerosolized will decrease when there's a presence of bacteria. And we test this hypothesis using ANCOVA model, and that indicate the significant effects on the covariate and also the variable. So for the discussions, our study suggests that the presence of bacteria will have a decreased impact on the number of aerosolized particles, and in particular the small particles. This is most likely due to the formation of the plastic agglomerates in the marine environment. And the study published by Summer in 2018 suggests that the marine bacterial glycoprotein exopolymer, EPS, plays a significant role in the formation of plastic agglomerates. So this marine bacterial EPS refers to the extracellular polymer substance produced by the bacteria in the marine environment, and they are made out of the complex molecules such as protein, polysaccharides, and lipids, and which this EPS will secrete outside of the bacterial cells and form a slimy layer around the bacteria. So the presence of EPS was observed to lead to the clustering of microplastic particles and will form the larger agglomerates. Since the microplastic agglomerates will become heavier, which means that there is a less likely to be carried by the air currents during the aerosolization process. So in conclusion, our findings suggest there are some potential impacts of MMP aerosolization. For example, on the MMP transport and exposure pathway. For the transport pathway, the smaller MMP particles are more likely to travel in the longer distance, which will result in the widespread dispersion and also the introduction into the new environment. For the exposure pathway, the increased aerosolization of the smaller MMP particles will increase the likelihood of inhalation of the particles by humans and organisms, which will have the potential issues on the respiratory system as well. Additionally, we have some potential impacts on climate process as well. So the result published from the paper published by Rafa in 2022, they found that the smaller polyethylene plastic led to an increase in the sea spray aerosol generations. And from this result, they hypothesized that the potential of the tiny powder polymers will enhance the sea spray aerosol productions, which will have the positive cooling effect on our atmosphere, since the presence of sea spray aerosol in the air will have several effects, such as to reduce the solar energy from the sun to reach the Earth's surface by the light scattering, and also act as the cloud condensation nuclei and ice nuclei, and also will influence the cloud formation albedo and precipitation cycles. However, this is just a hypothesis from that paper, and more research are needed. So for the next step, we suggest to test with other plastic polymers and shapes, since microplastic come in different polymers such as PP, PE, PVC, and also different shapes such as fiber, fragments, microspheres. And we want to investigate how different the chemical composition and the physical properties will influence the aerosolization process. Also, due to the time restriction, we don't manage to continue the experiments in the presence of microalgae using the mini-Mars systems. So we suggest to do that to explore the potential of the alga-mediated transfer of microplastics from the water bodies to the atmosphere. Also to explore the role of microalgae in the physical entrapment and the bioformation on the microplastics. So this novel perspective will help us to broaden our understanding about the environmental fate and transport pathway of microplastics in the future. Thank you for listening and attention. I want to take this time to thank my supervisor Anna, Martin, and Yunmeng, also all the lovely people for the help during my presentation and thesis preparation in advice. Thank you.
Thank you very much. Very interesting presentation. I will give the floor to you now. I would like to invite Simon Suter.
Okay, congratulations for the good job that we did. Just a couple of comments.
Yeah.
Well, one suggestion for also with microplastics in vertebrates. One is one of the compartments that you didn't mention in your introduction was part of the abiotic compartments such as water, seawater, sediment, and so on. Animal biotic.
Yeah.
Okay. Because they are also targets for pesticide accumulation.
Yeah.
Also, they will make a couple of comments. Okay, you used 5 microns polystyrene microplastics.
Yeah.
And you analyze the incorporation of these 5 microns polystyrene microplastics into microspheres.
Yeah.
How can you find microplastics of 5 microns in microspheres in sizes ranging 0.5 microns to 1 micron? If I'm not wrong, in one of your slides you show that there were microplastics in these very small microspheres.
Yeah, so for the trial experiment, because we just want to have a very quick look, so we only test one size, which is a 5 micrometers, because for the Rhodomonas salinas, we use this microalgae since it has a similar size with 5 microalgae as well. So it's better to look at how they interact together. So this is, because it's a trial experiment, so we just test one size, and we will just continue with these 4 size. And we use this 4 size because this is based on the previous study by the previous master students, so we can have a comparable results.
Yeah, almost reasonable.
Yeah.
What do you think? Well, these are pristine plastics. These are commercial ones.
Yeah.
Do you know that in the environment, primary plastics, they suffer changes according to the pressure of the environment due to the presence of ultraviolet light. So or changing of temperature or salinity or the action of fishes, crustaceans or mollusks, what you could expect from the microbiome in old or weathered microplastics.
You mean
Because you were already using clean ones.
Yeah.
And you are seeing that there are some bacteria attached.
Yeah.
But speculate, please.
So we start with this because we use the filter artificial seawater just to remove all the the extra the bacteria and algae inside to know the sole effects of this one, because we just want to know the interaction between these particles and these bacteria without any interaction with other the natural stuff. So we can know about this study first and to continue with the previous with the future studies.
You can see the point also because the work is not about to analyze the influence of global warming. I mean, discussing, you mentioned that this could help, this could be related, this could mitigate the influence of global warming in the seawater.
Yeah, this
How
is.
do you reach to that conclusion if you don't experimental design for that?
This is just a potential effect. That's why I didn't write as an effect. It's just potential effect because I read a paper about this just published recently, and in their discussion they also have this one as the hypothesis, and they just say that we need more discussion and more study about this. And so I just feel like it's interesting to add into my presentations and to have more discussion about this part.
Thank you.
Thank you.
Thank you. Yeah, collapse seeds, very nice work. I like a lot of microplastics. my work now, because we have a lot of microplastics. Have to study what is necessary. One question: why did we start with bacteria instead of microalgae?
Yeah,
because maybe microalgae are doing better sites.
Yeah, because we start with the smaller one. So because even microalgae have more significant impact, we feel like maybe we start with the bacteria and then we continue with the bigger microalgae, so we can have the very basic knowledge to proceed with the future study with the others as well.
Yes, because I saw that is the next step, no?
Yeah, next, yeah.
What type of experiments is necessary? Because, okay, we have microplastics in water, air, microplastics and why is important for human health, for ecosystem health? Why?
Yeah, because as my topic is about from the ocean to the atmosphere. So for example, if there's agglomerates, there's a potential effects maybe they will just agglomerate together, and then because they are nutrient biomes, maybe they will become slightly heavier, and then will be easier to be taken by maybe fish or something like that. Also, if there are the smaller particles released into the atmosphere, that will increase the potential to intake by human, or so this is related to human health as well. So I guess it's quite important to study these topics also with bacteria and microalgae, because this is all in our oceans.
Maybe it could be a good option to move because okay, now it's not in the air, it's in the ocean.
Yeah.
Maybe we can eat the fish that is eating the microplastics that we have in the ocean. So maybe it could be one option to do more density particles can be better. I don't know when. So maybe
Yeah. Thank you.
One option.
Okay.
So thank you. That's all.
Yeah.
Congrats.
Thank you.
Or after starts talking.
But I just have a curiosity. I think that all the methodology uses has been tested before and is and I wonder why you choose this flux of air that we have in this case and for 5 liters. Why is that? Is something related to machines, something environment?
Yeah, because if you look at this machine here, and this is a blank samples, and these are two replicates. Basically, because this is the only device in the world, and we just have 3 holes here to manage to put this collecting tube and to have more replicates because we need to run every time 24 hours. So to save the time and also to save the 6 liters of artificial seawater, we decided to have 3 new holes here and to connect to the filter holder. And also for the 30 liters per minute, because this is the maximum flow rate for the air cube here. So if we have 30 and divided by 4, each have the 4 to 5 liters per minute, and which is comparable with the trial experiment as well. Yeah, I guess this is some of the restrictions of the device because this is the only air cube we have in our lab. That's why, and this is the highest flow rate we can have at the moment. So we will start with this one, and this could be like study in the future with the higher flow rate as well. Thank you so much.
Do we have questions from the audience? Yes, Fiona?
Is there a certain reason why you choose those bacterial strains? Is it also because you were protocol this one?
You mean the
like why these bacterial strains?
You mean this one?
Yeah.
Yeah, because like before, during the preparation phase, we tried 6 bacteria that collect from the North Sea, and this one and the other one have the most jelly thing produced in the bacteria during the culture moment. That's why we choose this one because they have the more jelly thing to test the impact with the EPS with our bacteria and the microplastics.
Sure. Let's see, you've seen how microplastics can reach the air after being. There's a group in Toulouse which has analyzed microplastics in some high mountain catches in French Pyrenees.
Yeah.
One of their last research was in the an observatory, 3,000 meters. They did some analysis about the airs that were reaching the area. Some of them were supposed to come from Africa, another one from the United States. Do you think about doing something of kind of how many kilometers or something like that can those microplastics that are leaving the sea, the air, move? Because if they are only moving 1 meter, microplastics that are coming to the air in the open sea are not reaching the ocean.
Yeah.
They just can move like half a meter. Have you thought about it? Or?
Yeah, like for now, I don't have like the very exact and very solid thing to support this. But I guess because there are, for example, the 0.5 and 1 micrometers, they can be transported very long, and even like for the mountain and some of them, they just suspect there will be some source from the ocean, like from the ocean through the atmosphere and then to transport to the mountain and the other remote area. So I guess this is why we are doing this study, and because there's a very huge knowledge gap as well, so we need more study about this aerosolization process and how they would take this to the remote area as well. So,
you have time. One or just one question. Do you have any questions?
No.
What are your expectations for the step, or do you have any expectation of the next steps of the microbiology?
like I expect will be less aerosolized particles, since this is shown in our trial experiments, and maybe the microalgae, like based on different species, they could have more jelly substance, EPS, or even a biofilm to make the agglomerate together, because it will become heavier, even heavier than the bacteria microalgae agglomerates. So I expect it will be less aerosolized particle than a bacterial one. So,
Thank you very much.
thank you.
For this very interesting presentation.
Oh, no, I just want to.
Thank you for
Thank you.
the audience some insights of the background. Okay, you move to the third presenter.
Yeah, but we have like 3 minutes now if you want to start at 4. So we can wait or we can start right now, whatever you want.
What do you think, Alexandra? Like to wait?
I can wait 2 minutes. ¿Para acá? Lo voy a sacar.
Sí, sí. Déjame mirar.
Can I have a quick look at my presentation to see if it's...
Yeah, take the opportunity to move it forward and back.
But then I spoil. No, I wait, it's okay.
That's your choice.
It's no surprise.
I think we can start because there is a little bit late.
Yeah. Okay.
And it's way.
Okay.
So it's my pleasure to introduce the third speaker of this session, Alexandra Bettinelli, who's going to tell us about the use of microalgae for toxic metal bioremediation.
Thank you for the very nice introduction. Again, I'm Alexandra, and today I will talk about the use of microalgae for toxic metal bioremediation. My thesis was conducted at the Stazione Zoologica Anton Dohrn in Naples and in part at the University of Naples, Federico II. And I would like to take a moment to thank in particular my supervisor, Dr. Sergio Balzano, and my provoter, Professor Dr. Stefano Papirio, as well as all the other staff members in the Institute for helping and enable this thesis. So the focus of my thesis is on toxic metals which are released from industrial, agricultural and urban activities. They are highly toxic and non-degradable and hence threaten marine biodiversity and ecosystems in general. This problem is particularly present in the location of my thesis, the Gulf of Naples. Here we have 3 contamination hotspots. First, we have the Bagnoli Bay, which is the location of a ferrometallurgic plant, which you can see here in this image on the left. This plant was shut down in the 90s, but still releases toxins today. Then we have the mouth of the Sano River, which is famously known as the most polluted river in Europe. And lastly, we have the port of Naples, which is an international hub that receives over 10 million tourists and 50,000 ships a year. Taking together, this results in a high contamination of metals, and in particular cadmium, copper, lead, and zinc. One way to combat this problem is the use of organisms like algae, bacteria, and fungi to remove metals from the environment. Microalgae in particular can do this via 2 mechanisms. First, by biosorption, where metal ions bind to the cell surface passively, or actively through the transport across the cell wall and within the cytoplasm. In my thesis, I worked with Tetrasemus convolvulus, strain BS14, and this strain was isolated from the mouth of the Sano River and incubated for over 1 year in medium contaminated with micromolar concentration of metals. And this triggered a process called adaptive laboratory evolution, which led to an ecotype, Ale14, which showed a higher metal tolerance and higher bioremoval capacity than the original BS14 strain. You can see Ale14 here in this image on the left. And I worked with this strain and the experiments throughout my thesis. So my thesis consisted of 3 separated projects. So first, I focused on the isolation and identification of microalgae from sediments from polluted sites along Italy, mostly around Naples, but also some other locations. And we were interested in finding new candidates for bioremediation applications. Then I focused on Ali-14, which I mentioned before. So microalgae in general are non-axenic. So mostly they contain, apart from microalgae, also bacteria. And we were interested in seeing how this bacteria would affect the growth of Ali-14 and by this also bioremediation potential. So we conducted some co-culture experiments as well. And lastly, we tried to grow Ali-14 in a continuous flow system. Such systems are needed in order to allow scale-up applications and to treat real waste waters, for example, from industrial origin. So we were in this case interested in seeing if Ali-14 could remove metals as well as nutrients. Nutrients here were interesting because they're also a common anthropogenic pollutant, and we maybe have like a double beneficial effect using Ali-14 for such treatments. So I'm going to go through these topics one by one, starting with the identification of new bioremediation candidates. So first I received sediment samples from the locations marked in the map here, as you can see, mostly around Naples, and one a little bit further away in Mar Piccolo, or close to Brindisi. So I started by isolating these strains by micropipetting, then cultivated them and extracted DNA. We then continued to amplify and sequence the 18S ribosomal RNA gene. This is a highly conserved gene, which is usually used for identification studies. So we then continued to conduct a phylogenetic analysis, where we compared our isolated ATS ribosomal RNA sequences to known sequences in the NCBI gene database to find the highest match or the best identification of this species. We used those sequences and additional sequences to construct neighbor joining trees. So overall, we identified and isolated 14 microalgae strains, and we grouped them into 4 taxonomical groups. We had panda diatoms, centric diatoms, haptophyta, and green algae. Out of these 14 strains, 5 did not match with known species and probably make up novel species. To show you a little bit how I did this analysis, you can see here as an example the results for panda diatoms. You see on the left, we see the phylogenetic tree where our strains are marked in orange. And on the right, you see like microscopy images of the same strains. One thing you can see here, for example, is that T1 and T2, T3 and T5 show almost no evolutionary distance, like they show no horizontal branch lengths that separates them. So they likely make up the same species, but even though they have slightly different morphologies. We also can see that they do group with niche species, but they form a separate clade, indicating that they're novel species. This, if you compare this, for example, with BS26, this groups directly with Cylindrotheca clostridium and is likely identified as such. However, further research is needed to conduct a better or deeper morphological analysis using, for example, electron microscopy, or use additional genetic markers like the 28S ribosomal RNA gene. And I conducted a similar analysis with all the taxonomic groups. However, I'm just going to focus or show you this one due to the time constraints. But if you have questions about this, I'm happy to answer them later in the discussion. So moving on to the microalgae bacteria experiments. Like I mentioned at the beginning, we worked with a non-axenic Ali-14 culture. So we started by isolating bacteria and identifying them similar as before by sequencing the 16S RNA gene. And we identified 2 strains as Sulfidobacter and Alteromonas species. We then proceeded by making our culture axenic by incubating it with a mixture of antibiotics. And we used those strains together to conduct co-culture experiments. So we were first interested in seeing how different ratios of microalgae to bacteria would in fact influence the growth of Ali-14. So we started to test 3 different ratios, 1:10, 1:50, and 1:100 microalgae to bacteria. So here you can see the results are the growth curves of Ali-14. On the red line here indicates our control, which is an axenic Ali-14 culture. And the green lines here are co-cultures with Sulfidobacter, and blue here with Alteromonas. We did not find any statistically significant difference. However, we do see that there's a trend of a negative influence of the Sulfidobacter co-cultures and a slight positive influence of here the highest Alteromonas concentration. This indicates that there might be a species-specific interaction of microalgae to bacteria. So we then decided to continue with the second experiment using the highest bacterial concentrations. What I didn't mention, this experiment was conducted in F2 medium, which is seawater basically enriched with nutrients, vitamins, and trace elements. So we conducted a similar experiment, but this time we used a medium that was additionally enriched with toxic metals. Here we used cadmium, copper, lead, and zinc. And again, we wanted to see what is the effect on the growth of Ali-14. So you see again the growth cultures, the growth curves. Here in blue and green, if you can see, that's the co-cultures for Sulfidobacter and Alteromonas. And the red line and the light green line are again controls in axenic condition. So what we see here is that we seem to have a prolongation of the stationary phase compared to the control. And one common explanation for this is that at the end of the exponential phase on day 8, we have a depletion of nutrients in the medium. And bacteria can reintroduce nutrients into the medium by decomposing organic matter and thereby promoting the growth of Ali-14. We also see that there's not necessarily the same effect as before in the co-cultures without metals, so there might be effect of the presence of metals on the relationship of bacteria and algae. But this, of course, needs further investigation in order to identify the mechanisms involved in here. So lastly, coming to our continuous flow system, which you can see here in this scheme. It consists of 3 parts. So we have an influent tank, a photobioreactor, and an effluent tank. Our Ali-14 culture is here in the middle in the photobioreactor, which is provided with light, air, and stirring. So fresh medium is constantly introduced into the reactor, and at the same time, at the same flow rate, medium is removed from the reactor into the effluent tank. This flow rate is set so that the cell growth or the cell number in the reactor is maintained constant. In our case, Ali-14 has a doubling rate of 3 days. So the hydraulic retention time, the time needed to exchange the entire reactor volume, is also set to 3 days. In the experiment, we introduced metals again here in the influent. So the metals would then bind to microalgae in the reactor, which would be then removed to the effluent tank. In the end, we can centrifuge the effluent tank and thereby separate microalgae from the water. And in the best case, we remained with less metals in the aquatic solution than before. The actual setup looked like this. So you see here again, influent tank, photobioreactor, and effluent tank. And the experiment overall lasted 45 days, and we tested 3 different conditions. We first had a negative control, where we did not introduce any metals into the system. Then we had low concentrations of the same metals as before, cadmium, copper, lead, and zinc, and then a higher concentration of the same metals. Like I mentioned briefly in the beginning, that we also looked at nutrients because they're common anthropogenic pollutants. So we were first looking at how nutrients would be removed from the medium. And we found that there's over 65% of nitrate removal and over 99% of phosphate removal. Here these plots show the influent concentration in white and the effluent concentration in black for the both metal concentrations, low and high. And one thing you might notice if you look at this graph, if you look at phosphate, we seem to not have the same influent concentration of phosphate. And this could be because phosphate might react with metals. So in the condition with higher metals, we might have slightly more phosphate reacting and precipitating and be removing from the dissolved phase. So that could explain this difference here. We also see that the nitrate removal here is not complete, and this could be improved by prolonging the hydraulic retention time and the time the microalgae have to absorb the nitrates. So finally, coming to the metal removal in our system, you can see here the results for cadmium on the left and for copper on the right. Again, these graphs here show the influent and effluent concentrations in the different conditions. And we also calculated the percentage difference, the difference between before and after the treatment, basically, between influent and effluent, which you can see in the table above. One thing you might notice looking at this graph is that we seem to have a higher concentration of metals in the control, where we didn't add any metals, compared to the low and high concentration. We also see that we have quite high error bars in all the conditions. And this is probably because we were at the lower detection limit of our instrument. We used here atomic absorption spectrometry, and we probably had a sensibility error. We also had to recalibrate the instrument each measurement, and since we did not measure everything in one day, we had slightly varying standards, making the results very difficult to interpret statistically or generally. However, we can focus on the highest concentration, which we found to be the most informative. And we do see here that we have an 80% removal of cadmium and 30% removal of copper. For copper, we had some significance, but still, considering the high error bar, we have to be a little bit careful by interpreting these results. We had similar findings for lead and zinc, and in this case, we also just focused on the high concentration as we considered this the most informative. In both cases, we did obtain a removal of over 50%, indicating that the species indeed is useful to remove metals from aquatic solutions. However, again, high error bars, so this indicates that we need to improve our analysis or switch to a different technique like ICP-MS, which is a type of mass spectrometry. And this was originally planned, but unfortunately, we didn't have availability of the machine in the end, so we used an alternative technique. So, to conclude, just quickly, what does this thesis say, basically? So, we were able to identify new species from polluted sediments that are probably useful bioremediation candidates. However, we do need to test further their metal tolerance and and select the best candidates for further bioremoval studies. We also found that Alteromonas and Sulfidobacter species are associated to our Ale-14 culture. And we showed that the consortia balance is affected by the species, identity of bacterial algae, the ratio they incubated, and the presence of metals. Lastly, we showed that Ale-14 is able to grow in a continuous flow system and simultaneously remove metals as well as nutrients. This is key because most real wastewaters consist of a mixture of pollutants and not only one. So Ale-14 would be very promising to treat, for example, port waters, which are usually very enriched in nutrients and contain a mixture of metals as well as other pollutants. An alternative application for this would be the treatment of indoor aquaculture wastewaters, which are also highly enriched in nutrients. So if you're interested in this topic, here are some of the references I used for the presentation and the thesis. And thank you very much for your attention. I keep spinning.
Time for questions. So to keep the balance, I will try as long as I can. Thank you, Alessandra. Congrats.
Thank you.
Now is the presentation. Now only some questions or maybe questions. It's a very interesting work, and I have questions, but it's not the moment. Maybe I can send you an email or something like this.
Yeah.
It's an interesting work for my tour easily.
Okay.
Here. Why Ale-14? You have some references to so about it.
Yeah, like this algae was isolated before in the laboratory at the Stazione Zoologica, and they also tested the metal tolerance of the strain. And it's also because we work in Naples, and it would be better if we try to treat the contaminated water in the Gulf to use an endemic species, like a species from the place, and not introduce a new species which could potentially, in any case, get free and contaminate, or not contaminate, that's not a word. Yeah, be reintroduced in a foreign environment. Yeah. They are very tolerant because they are directly isolated for sediments that already contain high metal concentrations. So they're already adapted to it. That's why we work with those isolated species.
Maybe forget it, but how to find this microalgae in the first experiment?
Similar as the one before, as the isolations that I showed at the beginning. Oh.
The first experiment that we developed with techniques are
this here basically. It's the same procedure, but it was not for Ale-14. It was not conducted by me, but by previous students and the professor. But yeah, you receive sediment samples basically, and it's a little bit like the picking of the microplastic. You have a fine tube and you start picking the microalgae and try to get a clean culture that only contains one species. And this is then cultivated until it is clean basically. And then we conducted sequencing and identification. Yeah. So, but Ali14 was already published in a paper last year, so it is already proven to be correctly identified.
And now it's like in your house.
Yeah.
What about the next step?
Yeah.
Understand the next step in the final slide, but if you have to start a new experiment, more experimentals from one very good area, what do you do?
I mean, I started a lot of projects, so I think I can work on all of them in more depth, I would say. So for microalgae, for the isolation, for example, we do need, in order to publicate the results of these isolations, we do need to conduct electron microscopy, which was also not available because the machine was not available. And we need to conduct a further genetic analysis because the 18S is a very good indicator for the species, but for microalgae, it doesn't have a high enough resolution necessarily to be very clear about the identification. So to be sure about this, we do need to conduct those steps further.
For the another genes in the database.
The what?
Other markers in the database.
Yeah. For microalgae, they use the 28S ribosomal RNA, which is from the. The 18S, it's the usually one. It's the most conserved one or the most known for this. But there are some publications that indicate you need to conduct further research with other markers. That's one of the results I didn't show, where we looked at pavlova species. And they are also very difficult to just identify with the 18S because they sometimes group species together even though they are not necessarily from the same clade, let's say. So in order to be sure about this, but you don't have to. It's a suggestion to just be more sure about results, let's say. Yeah. Or genetic, also gene like protein expressing genes would also work. Yeah.
Thank you. Congrats again. Just have a small congratulations and all those are related removing contaminants from the environment. I only have one curiosity. You presented on a slide where you showed the impact when you added and you have already difference in where this side and in
the effluent it's here. It's just not existent.
The thing is, because you have just a curiosity, I don't know if it can happen or not. It's possible because you have classification of there's more classification.
That's true. Because we separate them by centrifugation. So we have precipitated metals, for example, and phosphate together. It might change the results.
And it can be something that stay not just in the but in the tank reaction tank.
That we introduced actually less metals in the medium. The thing is, the medium it's prepared in advance, so it's not like we redo the medium constantly. So if there's participation, it will probably, I believe, it will likely be in the influent tank. Like it would already precipitate here. So we introduce less.
The idea is if it precipitates at the beginning. It cannot precipitate while
it can be,
It
can happen. It can happen. I'm not sure about this because we didn't notice this until we saw the phosphate results, basically. And then I did some research that indicate that there might be a reaction between the different metals.
Is everything at the beginning of the experiment, or am I keeping things and collecting that?
That is possible. Though the loss is not. Oh, sorry, it's here. I mean, the scale is also micromolar, so we have a loss of, let's say, 3 micromolar of phosphate. So it might be not a huge difference. That's a little bit, but yeah, that's probably what we need to investigate also. How it, yeah, but that's
It's curious because I don't know if it's possible.
yeah. But this is probably then we have to improve the medium we work with to avoid that it precipitates. Because in the ocean, that's like the seawater in itself has also very, very low concentrations of metals normally. So in this case, we don't have precipitation, but metals are rather bound to the sediment also. Yeah. But yeah, for the experiment, that's yeah, for sure the drawback.
Very nice.
Thank you. Congratulations. You did well. There was some curiosities as always. So the influent tank, if I'm not wrong, algae are present together with.
No. The influent is just seawater, basically. Seawater, which has nutrients, trace metals, or trace elements, and vitamins. And all the algae are here, basically. And here's the reaction. And like I said, the rate is set slowly so that here we have a constant growth. Like if they double every 3 days, the reactor volume needs to be removed.
So you detach the metal from the surface of the algae.
They should not detach. They should detach. They can be detached by acidifying the solution, but they're usually bound to different mechanisms, which I did not explain in detail, but there are ionic bonds, there are covalent bonds, different kinds of bonds. And since we work with living algae, they also introduced within the cell.
If you change the redox conditions, you change the link to the algae and the metal.
There are various mechanisms. Or what do you mean? I'm not sure if I understand.
In the tank, in the photoreactor,
yeah.
algae together with metals.
Mm-hmm.
So they are absorbed to the surface of the algae.
Yeah.
According to those conditions present in the photoreactor.
Mm-hmm.
How you detach? How do you release?
How to recover again the metals?
Because you can release the metal and take the algae.
Yeah.
Never-ending.
Yeah.
Capability of reducing the amount of metals.
Yeah, but that's actually one limitation of such systems that is still not resolved. How to recover the metals, because metals, they do not disappear; they are still present there. The difference is that we reduce the volume we have. Like in this experiment, we centrifuge 24 liters, and in the end, you end up with a gram of microalgae. So you have it very highly concentrated. And I mean, there's different approaches to this. Also, how to harvest cells in the end. Like we did now a free suspended system, but other systems use, for example, algae bound to sodium alginate or to other matrices, basically. This has a little bit of problem that the algae are dead or apparently growing very, very slow. So you lose this accumulation process, which is also high contributing to the removal. But yeah, and centrifugation is one good method to separate them and just remove them. And then you still have to deal with the end product. And you potentially can reuse the microalgae for biofuel for other procedures.
Because the metal is not coming into the algae.
Yeah.
It's not crossing
the
cell wall of the algae onto the.
They do cross. But what you can do, for example, if you work different species, for example, produce high amounts of lipids. So one idea is to extract the lipids, use those for biofuel production, and then you remain with a tiny amount of metal still, and you can remove them safely, basically.
What can happen if the developments in the real environment are very high, be toxic for the algae?
Yeah, but we work with concentrations that are much higher than what we found in the ocean.
They're much higher.
They're higher, yeah. And the lower the concentration of the metals, the better the removal capacity, actually. So it's when they're full system.
Do you think that you can apply this into the environment?
Yeah, I hope so.
Good luck.
The problem is that actually the recovery of the algae. Yeah, because centrifugation is very powerful. You need a lot of electricity. That's expensive. That's not feasible. But yeah, that's why there are different approaches, and there's a lot of research on this that use different types of reactors and everything. This is a very simple start, but it was the first one that was conducted in this experiment. So just
also in apples
they use bacteria also a lot.
It's fungi.
For what?
Reducing the electronic raw materials.
Oh, okay. Like in the lab I worked, they used bacteria as well. That's why I conducted this co-culture experiment. It was a cooperation between my group and the bacteria group, which also works in bioremediation, but based on bacteria, basically. Yeah.
Thank you very much.
No, you're welcome.
Do you have questions from the audience? Yes.
Environment and
Hmm.
the products habitat when they are digitized and
No, it's always a contaminant. Like it stands, it's not getting transformed. Like it does get transformed, it binds, but I mean, especially microalgae, they are consumed by shellfish, and it goes up into the like biomagnification, bioaccumulation. That's the problem. And higher concentrations do tend to be very toxic. Low concentrations, not. For example, zinc is an essential element, so we do need it in low concentrations. Yeah. But when, for example, if the cell dies, it either can be, yeah, it sinks, like it's more concentrated in the sediment for sure. That's also why there's little data on concentration in seawater, because it's very influenced by like water turbulences, by river input, by all these factors. But in the sediment, you can measure those concentrations quite stable because they just accumulated, and then they are obtained again or reintroduced in the trophic net by organisms. So yeah.
Do you have more questions? Yes, no?
It's difficult to maintain the transport system.
Yeah. I have an image on this actually, if you're interested. Not difficult. They are very stable, so they recover quickly. This is the growth curve I had actually. Like I measured, cell number by flow cytometry and also optical density, which then lead to TSS, the total solids, suspended solids. And this is the growth variation throughout the 45 days. So you do see that there are quite some ups and downs, but I mean, they are varying about 1 million and 2 million cells per milliliter. So it's not a huge, but yeah, you do have to calibrate the system a little bit. And also there are some factors that do affect cell growth quite quickly. For example, the thing marked here, the black out, there was a power failure in the night, so it would not be removed from the effluent, and then cell number just peaks. So yeah. But they are robust, so they recover quickly.
I have a very quick question. Are there questions from you? If not, I hope you recovered from your audio state. Thank you
I
very
hope
much
so too.
for the presentation. Thank you very much for all the three fantastic presenters of this session, for the participation of the audience. Now I invite you to the coffee break, and we will resume again at 5 o'clock.
today because we have three presentations, and we are going to say, well, first I would like to present the Trinidad Pérez and nosotros and what we entered. Okay. And we are going to start with Natasha. Okay. You remember you have 15 minutes for the presentation. I will let you know the last 5 minutes. So I will just have my hand to let you know. And then there will be for questions. Okay. So,
okay, good afternoon, everyone. My name is Natasha Melibukowicz. Today, I will be talking about the spatial and temporal evolution of fatty acids in the South Senegalese Upwelling Center. I completed my master thesis in Brest, France, where I was supervised by Drs. Philippe Sidon, Dr. Eric Machu, and Dr. Gauthier Schell. So first, we need to understand what are fatty acids. So fatty acids are an important energy source for all marine species in the oceans, and they make up the structural components for cells and cell integrity. They're also essential for growth, reproduction, and larval development in higher trophic level organisms, and this also extends to human health, where we require certain fatty acids for neural development and growth. What's interesting about essential fatty acids is that they're synthesized almost exclusively by phytoplankton, where they make up 5 to 60% of cell dry weight. Here you can see what's known as long-chain polyunsaturated fatty acids, also known as LC-PUFAs for short. And these are the essential fatty acids that are synthesized by phytoplankton predominantly. You can see for simplicity the short form here, EPA, DHA, and ARA of essential fatty acids. And you may also know them as omega-3 and omega-6s, which are the same fatty acids that we acquire from our diets through the consumption of seafood. So we need to understand a little bit about phytoplankton and how they synthesize fatty acids. So phytoplankton make up the base of the pelagic food web, composing the suspended particulate organic matter. Phytoplankton is responsible for 50% of global primary production, and like all photosynthetic organisms, they're highly controlled by light, temperature, and nutrient availability. So in the context of tropical regions, nitrogen is the nutrient availability here. What's interesting is that species of phytoplankton have various growth rates, tolerance to changing conditions, and as well fatty acid profiles. So as you can see here, these are the major phytoplankton groups and their components of fatty acids. So you can see as an example, diatoms have a very high proportion of EPA fatty acid, whereas comparing to dinoflagellates, they have a very low proportion. This means that the phytoplankton community composition in your ecosystem will impact what fatty acids are available to be consumed by higher trophic level organisms. So I get asked all the time, why are you studying fats in the ocean? The first reason is to understand the source of organic matter. So this will tell you what fatty acids are present in the ecosystem. It's important to know what fatty acids are available at the base so you can understand what's available for the whole ecosystem and the productivity. For example, DHA is higher in proportion to dinoflagellates, and EPA is higher in diatoms. Next, fatty acids are used to understand trophic relationships. So out of all the fatty acids available, there are known biomarkers of species that have been documented in literature, and this can aid in trophic ecology studies. So for example, if you study the fatty acids in a specific species, you can understand who is consuming who, and then you can start to create and build a whole trophic web of an ecosystem. Lastly, we can understand ecosystem quality. So LC-PUFAs that are synthesized by phytoplankton contribute to a high-quality ecosystem. And this can provide information on the quality of food sources in important ecosystems, especially ecosystems such as eastern boundary upwelling systems, also known as EBIS. What's interesting about EBIS is that they cover 5% of the ocean surface, yet they account for 25% of the world's fish catch. This is due to upwelling, where surface winds take surface water offshore, and then they're replaced by deep, nutrient-rich cold waters that come to the surface. And these high nutrient, cold, dense waters instigate phytoplankton blooms, which then create the food supply for the rest of the trophic web. Here, you can see the 4 major EBIS of the world: California, Peru, Northwest Africa, and Benguela, and you can see chlorophyll A levels mapped here. And you can see the high chlorophyll on all of these eastern boundaries. Specifically looking at Northwest Africa, you can see the South Senegalese Upwelling Center, also known as the SSUC. So this is quite specific to other EBIS because it has shallow continental shelf of 20 to 30 meters depth, and that stretches for a long space of time. And it is sheltered from mesoscale influence, so you can see here, the sheltering causes an upwelling direction from a north to south direction. So this map is showing SST, sea surface temperature, and you can see the cold plume from north to south. This area is unfortunately exposed to a lot of regional threats, the major one being overfishing. There is a lot of documentation of low fish stocks in the area. And upwelling favorable winds are especially sensitive to climate variability. So we know that the changing in the winds is going to cause potentially a change in phytoplankton community composition and fatty acids available. It is expected that globally there will be insufficient omega-3 by the year 2050, and this is most likely due to overfishing and as well because of climate change. Currently in the SSUC, there is no fatty acid data in this region. So it is important to fill this gap to understand a baseline of fatty acids in this area here, which is on the coast of Senegal, just south of Dakar, and hopefully to compare to other EBIS and see how they compare and contrast. So my study was to determine the fatty acids in the South Senegalese Upwelling Center. First, I wanted to look at what is the quality of long-chain polyunsaturated fatty acids, also known as LC-PUFAs, in a post-upwelling water mass. So we predicted that LC-PUFAs and nutritional quality will significantly decrease over time and at depth. Next, we wanted to characterize the aging of a water mass using fatty acid biomarkers. And here we predict that bacterial fatty acid biomarkers will increase over stations and depth as the water mass ages. To do this, I collected samples on the SCOPES campaign, which was a 25-day campaign on the South Senegalese Upwelling Center, which you can see here just south of Dakar, Senegal. There were 46 stations where fatty acids samples were collected by collecting water from Niskin bottles, filtered in triplicates, and stored in the freezer until analysis back in France. We also collected environmental data from the CTD, temperature and nutrients, and we also looked at chlorophyll A and accessory pigments taken from a fluor probe seen here. So briefly, just to go over, how do we quantify the fatty acids in our samples? So through a process of extracting the lipids from our samples and what's known as saponification and transmethylation, you end up with what's called a FAME, or fatty acid methyl ester. So this fatty acid methyl ester just means that the lipid has been transformed into a volatile form that can be read by gas chromatography. So once the sample is analyzed, you end up with what's called a chromatogram, where each peak represents a different fatty acid in your sample. Then you can compare it to in-house standards and in-sample standards, and you end up with the concentration of all fatty acids found in your samples. Moving on to the results, we first needed to characterize our water mass. So here you can see we did this by doing a Lagrangian experiment. A Lagrangian experiment just means we wanted to make sure we were sampling within the same water mass at all times. To do this, we followed the trajectory of an SVP buoy. You can see here the color resolution is not as good using the screen, but I promise you can see it is like this orientation. And basically what this did was we allowed the boat to follow this trajectory of the SVP buoy to make sure we were maintaining in the same water mass at all times. So in typical upwelling, we would expect to see the SVP buoy travel from a north to south direction, like I mentioned. However, it only just made a slight up and down, meaning we were not in true upwelling, but a post-upwelling condition. So based on this, we selected 6 stations, station 20 to 29, for fatty acid analysis. And this happened over a 3-day period, morning and evening. 2 to 3 depths were selected, depending on the depth profile, where there was surface, oxygen maximum zone, and oxygen minimum zone sampled. It's important to know that the OMZ, or oxygen minimum zone, is not a true OMZ, as this is a very shallow shelf, but for categorical reasons, it separates the 3 depths. Next, we looked at some of the environmental data. We looked at all nutrients, temperature, pigments, etc. But here I'm highlighting the temperature and the nitrates. So temperature was low variability between 19 to 22 degrees, so it did not change very much, but it was similar to the upwelled water temperatures. Nitrates, you can see, are quite low at the surface, and of all nutrients, nitrates were the most dominant, and they're dominant at O2 max, with quite variability. And this is shown over stations, at the surface, oxygen maximum, and oxygen minimum zones. This is indicative of high productive areas where you have the nitrates influencing phytoplankton blooms. Next, looking at chlorophyll for the same depth profiles, you can see that it's decreasing at the surface from station 20 to 21 to 29. This makes sense because there is low nutrients here, and phytoplankton are not able to grow under low nutrient conditions. Next, looking at the fatty acid data. So out of all of the 55 fatty acids we found in our samples, we selected the top 26 fatty acids that were above 1% of total fatty acid proportion. And here we did a principal component analysis to see if we could find relationships of biomarkers of our fatty acids. So we expected to see a lot more relationships, but of the ones we found were these C16 polyunsaturated fatty acids in the negative side of axis 1. And you can see that they make up a high contribution of the variability in our data. Moving on to the oxygen maximum zone, you can see there is a cluster of ISO15, ISO16, and anti-15, which make up the bacterial markers fatty acids. So these were the ones we decided to explore further. Here you can see diatom and bacterial fatty acid markers we saw for all stations at surface, oxygen max, and oxygen minimum zones. So there were significant differences between our stations, but there was no clear trend that we could path over time, which is stations, or through depth. At the OMZ, there is an increasing trend, although we do not have enough data to make significant interpretations here. Bacterial markers, we had significant differences between our stations as well, but again, no clear trend over time. However, there was significant increases with depth, which is what we expected to find, because at depth there was lower amounts of phytoplankton, and that's being replaced by bacterial activity. In conclusion of these results, the amounts of diatom and bacterial markers does not clearly characterize an aging water mass, which is what we expected. We expected the bacterial markers to increase over time and the diatom markers to decrease, but this is not what happened. This is likely due to the high nutrients that are still available through time here. And lastly, we looked at the nutritional quality index to see the quality of fatty acids in our system. So this is calculated using this equation here. Basically, it factors in the LC-PUFAs we were talking about, DHA, EPA, and ARA. So at the surface, there was significant difference from station 20 to 21, but again, no clear trend over time. At the oxygen maximum, however, there is significant increase in trends over the stations, which means it is increasing over time, as well as at the OMZ. So this contradicts our initial hypothesis. We thought the nutritional quality would decrease over time as the nutrients are consumed. But again, our experiment shows that nutrients were remained high over time, and this most likely increased phytoplankton production and increasing the available LC-PUFAs of high quality. So most likely we need to compose a longer study, one that would actually show true aging water mass, because the nutrients do not show that. What's interesting is that values are similar to those found during an upwelling event in a Benguela upwelling system. So this is comparing to another EBIS. And this is interesting because we are in a post-upwelling condition here, but the values of NQI are similar to true upwelling. So we are still missing a lot of data from this area, so it would most likely be interesting to do a true Lagrangian experiment within an upwelling event in this area, as well as post-upwelling, and then compare it to other EBIS. In conclusion, our environmental data and Lagrangian SVP buoy indicates post-upwelling conditions. 16 carbon PUFAs, which are the biomarkers of diatoms, composed a moderate amount of the total fatty acids at all depths, and they correlated to brown algae that was measured using the fluoroprobe. Essential LC PUFAs and NQI increased over stations, while bacteria fatty acid markers generally decreased. So this was in contrast to what we expected, but again, most likely due to the high productivity still happening in this 3-day experiment. Nonetheless, this study provided novel fatty acid data that is not currently available in the SSUC, and this is important for characterization for eventual ecosystem-based management. Thank you for your attention.
So now
I'm just good. Okay, thank you very much for very interesting approximately. So I wonder after all the capture what is the next step?
That's a great question. So I'm currently did more lab work to analyze samples from other stations in this because I did collect on board 46 stations, but I only analyzed 6 of them. So I think the 3-day period was good for our master thesis, but I would like to include other stations available maybe outside of the upwelling zone comparing to inside of the upwelling zone. We also have zooplankton fatty acid data that we would like to look at. So maybe comparing that to look at biomarkers found in both and maybe start to make connections there.
That's helpful.
Okay. Congratulations for the work. What was your implication in the work? Did you go to the campaign?
Yes. Yeah, so I was on the campaign in December 2022. It was 25 days and we were on the coast of Senegal. It ended in January 2023.
Also, you did the analysis in the lab?
Yes.
All the results you obtained?
Yeah, I did all of the analysis. Obviously with help from my lab mates and people, but yes, I performed the analysis.
Another question is related with the title that spatial and temporal evolution of fatty acids.
Yes.
I see the spatial one because you have something in different stations and you space.
Yes.
You obtain different results and you make different predictions about the distribution of fatty acids among the parameters.
Yes.
But what about the temporal evolution? You didn't, as far as I know, say with the report.
Right.
Just only looking at your presentation.
Yeah.
Do you mean temporal, seasonal? There are different times. What do you think? Increments of time. There are different seasons that could affect, of course, the presence of diatoms or dinoflagellates.
Right. So we don't have long-term temporal measurements because this is the first time we do measure fatty acids in this area. But temporal is the fine-scale temporal. So from station 20 to station 29 happens over a 3-day period. So 3-day period, yeah, to follow the water mass. So it's more of a fine-scale temporal evolution that we're looking at.
You think that this 3 times period is enough to obtain?
No, I think we need definitely a longer duration of experimentation. But it was for the purpose of we had the measure of where the boy was, and we only had that availability for the 3-day experiment. And that was organized from the campaign. So that Lagrangian experiment encompassed just a 3-day period. So I had to follow that timeline.
Okay.
I have a couple questions, if that's okay. Actually, just what you said right now, you walked off the ship with your samples in January
2023 is when the campaign ended.
And you finished by just in June.
Yeah.
That's an amazing amount of work. I'm really so congratulations.
Thank you. Thanks.
I didn't quite understand from your figures where you had depth.
So I will show you. So just as an example, our depth was we sampled 2 or 3 depths from using the rosette from the Niskin bottles, and the depths were selected based on the CTD profiles on board. So at each station, the depths were slightly different. So in order to intercompare between stations, I assigned them categorical. So that was surface, oxygen maximum, and oxygen minimum zones.
And where are they columns in this and the following?
So
Here, yeah.
Right. So I don't compare. I always separate all 3 depth profiles. I don't compile them.
Okay.
So in all parts, they're separated. Yeah.
And then I'll follow up by asking the choice of fatty acids. So you just talked about
yes.
Because you can't have gazillion of them. But what if the markers or most significant biomarkers are actually rather rare ones.
That's very possible, and that's true.
And do you have any?
So
Did you mention that in the literature or discussions in your lab?
yeah, so I definitely worked closely with my supervisor on that because he has really good expertise on the history of biomarkers and the best ones to select. So in general, the ones that we like to look at are in higher abundance, above 1% of total fatty acids. But his expertise would we would decide if there was any that we should keep in the sample set to make sure. But in our case, in the data we had, no, there was none under that were in trace amounts that we were interested in looking at least for suspended particulate organic matter. Maybe if you were looking at in zooplankton or in small pelagic fish, there would be some of those trace ones you would keep, but in my case, we did not.
Okay, so my one more has to do with the Lagrangian and also the that you go out there, you want to know what I'm.
Yes.
So I appreciate that very much. But
yes.
If you were in a Lagrangian experiment in an upwelling, isn't there water mass always going like this and being spread?
Yeah.
So that you can never really sample the same water. You can't follow a water mass.
Right. So what happened is in this area specifically, upwelling happens from north to south. So we were expecting, if it was an upwelling water mass, that it would travel from north to south. So this SVP buoy is what we put in the water so that we could make sure we were in the same water mass, or we hope that we are in the same water mass, and the boat would follow the trajectory of this buoy. So we were always trying to maintain within the same range of this buoy at all times.
We're supposed to wait because people online.
Online might be joining.
All of
You
your
can see,
friends.
you can
think
see
they-
there.
Yeah, no worries. I'm gonna go get my water.
Excuse me. You fix it so that it is on the screen. Because here, see how it was off the screen. You know what it checks? Is there not a automatic thing that with arrows?
I think it should work with the graphs and stuff. So good afternoon, everyone. My name is Julie, and for my master thesis, I spent 5 months at the Irish Whale and Dolphin Group in Ireland to assess the risk of whale entanglement in pot fisheries in Irish waters. So what exactly are pot fisheries and how are they related to entanglement? So pot fisheries are fisheries that use baited basket-like traps to specifically catch shellfish, such as crabs and lobsters. In a typical setup, pots are connected to each other through a ground line with short leg lines, and each of the endings are connected with a vertical rope to a surface buoy. They are put on the seabed for a couple of days before being hauled back up. But the problem with these fisheries is that they use buoyant ropes. So a lot of rope ends up in the water column and at the surface, and this is where they can pose a threat to whales. Entanglement is an increasing issue worldwide. In the North Atlantic, it's even the leading cause of mortality for large whales. Consequences are obviously death and through drowning, but also increased stress and injuries that can lead to infections and impaired ability of swimming and foraging. So what's the situation like in Ireland? In Ireland, 85% of fishing activities are located inside territorial waters, so inside the 12 nautical mile area. And this overlaps with the feeding areas of a couple of large whales that are present in abundance in Ireland. There are indications that entanglement is happening, but at this moment we don't know the extent of the problem or even where the areas are located where this entanglement can happen. So what were the aim of the thesis? My aims were to create distribution maps of the 3 most abundant whale species in the Irish EEZ. This is for the humpback whale, the minke whale, and the fin whale. We then also looked at identifying areas with high fishing activity to then determine the co-occurrence of whales and pod fisheries to identify high-risk areas. And then lastly, we were going to look as well at the strandings data and see how this relates to cases of entanglement and the location of the high-risk areas. So the study area was the Irish EEZ, or Exclusive Economic Zone, that extends up to 200 nautical miles off of the Irish baseline. It includes the Atlantic part of the Atlantic Ocean, the Irish Sea, and the Celtic Sea. So for the data collection, we looked at 3 different types of data, sightings data, fisheries data, and strandings data. For the sightings data, we collected over 100 different, or we looked at 100 different data sets of effort and non-effort related data over a period of 18 years from 2005 to 2022. All the opportunistic data in Ireland is reported to the Irish Whale and Dolphin Group and is also quality controlled whenever a sighting is reported. The fisheries data was obtained from the Marine Institute of Ireland and consisted of 2 different types of data, VMS data and landing records data. The VMS data, or the Vessel Monitoring System, is a system that records and transmits the geographic location and the speed of a vessel, and this for vessels of 12 meters and larger. And based on this information, we can determine how much time a vessel has been fishing. And so in this case, the Marine Institute provided us the fishing effort for all these boats of 12 meters and larger over a period of 5 years from 2014 to 2018. But because this data is only available for vessels of 12 meters and larger, and we're interested in obviously the whole Irish fleet, we also looked at landing records. Vessels of 10 meters and larger have to report their landings in logbooks at ICS rectangle level, which is a gridded area of the Atlantic Ocean that's been developed by the International Council for the Exploration of the Sea and are basically big areas of 30 by 30 nautical miles. Vessels below 10 meters, however, are not reporting this at ICS rectangle level, but do report this on slips and sails notes at the harbor where the sail occurred of the landing. So in this study, we assumed that the ICS rectangle where the harbor was located and the sail occurred was also the same ICS rectangle that the boat went fishing in. And the Marine Institute then basically calculated with this information the number of bots that were present per ICS rectangle for the 2 biggest fisheries in Ireland, the lobsters and the brown crab fisheries, and this for the year 2022. And then for the strandings data, all the strandings get reported also to the Irish Whale and Dolphin Group. And whenever a stranding is reported, a member of the strandings network goes to the location to get basic information on the stranded animal. And we looked at a period here of 1990 to 2022. All the data analysis was performed in QGIS, and we performed like density maps for the different types of data. We overlaid a 10 by 10 square kilometer grid for the sightings data and the boats of 12 meters and larger to assess the density. And then for the number of pods for the whole Irish fleet, we only looked at ICS rectangle because they were reported at ICS rectangle. So for these, we calculated the pod densities at these grid size. And then for further analysis, we basically overlapped the whale density data and the fisheries data at the same resolution. So depending on which type of fisheries data we were using, we were adjusting the whale data. And then in each grid cell, we would basically calculate, or with an index of co-occurrence, how likely it was that the pot fisheries and the whales were co-occur in the same grid cell. And this is also called the risk of entanglement measure and was developed by Northridge et al. And so here we look at basically how the whale density would vary from the mean whale density and how the pot density would differ from the mean pot density to, in this way, calculate the index of co-occurrence and have an idea about the risk of entanglement. So going over to the results, here on these maps, you can see the whale density data for the three most abundant species, the humpback whale, the minke whale, and the fin whale. And these are reported as number of individuals per square kilometers at a grid size of 10 by 10 square kilometer. You can see that for the minke whales and the humpback whales, the highest densities were reported of the southwest coast and the south coast, while for fin whales, this was mostly of the south coast. Highest densities were reported for the minke whales, followed by the fin whales, and then the humpback whales, which is also what's been reported in previous literature. So for the fisheries data, we also looked, you can see here these two maps with, on the left side, the map reporting the fishing effort for the boats of 12 meters and larger. And this has mean fishing hours per year at a grid size of 10 by 10. And then for the pot density for the whole Irish fleet, it is reported as number of spots per square kilometer, but at a bigger grid size, as you can see, this is the ICS rectangle level. And the main thing that we can see here is that for the boats of 12 meters and larger, we don't see any fishing effort off the south coast or north of Dublin on the east coast and of the northwest coast. While when we look at the ICS rectangle level, we see that there is, well, pretty much spots everywhere along the coast. And so when we were going to look at the risk of entanglement measure for the boats of 12 meters and larger and each individual whale species, here on these maps you can see the black dots, which are areas where basically pot fishing was reported, but no whales were present. So the REM value was 0. But we can also see that off the southwest coast, we have a very big or very high risk area for all the three whale species. For the fin whales on the right side, there is also an additional risk area of the southeast that can be observed. And then when we did the same assessment, but for the whole Irish fleet, there is additional areas that are popping up off the south coast. But this is obviously because we have additional data on the whole fleet in this case, and not only on the boats of 12 meters and larger. So this highlights the fact that obviously we need information on all these different boats and the pot fisheries. But also something that needs to be addressed here is the fact that we're looking at a very big resolution. We're looking at ICS rectangle level, which are areas of 3,200 square kilometers. So here the chance is that we could overlook areas where there is a very high number of pots, but that are not popping up on these maps. And then when we looked at the strandings, we didn't see any significant increase over the last years in the number of strandings, and also the number of entanglement-related strandings were only reported as 9 cases. But we do also know that there is a big underreporting happening in Ireland because of a number of reasons, such as a very long and broad coastline, but also a very inaccessible one. Also specifically to entanglement, when we looked at historical data, it was hard to be able to identify if we could see rope marks or not, because the photographic evidence or the quality of the pictures was not high enough. And also in general, no post-mortem analysis are being done on the stranded animals to identify whether or not entanglement is an issue. So basically, this study was able to identify a couple of constraints in the current analysis, specifically for the fisheries data and the entanglement-related data. As mentioned, we looked at the fisheries data from lobsters and brown crabs, but there is 6 other fisheries that are using pots in Ireland that we couldn't look at because we didn't have the data. Also, we did have information on the whole Irish fleet, but there is obviously also international vessels that are fishing in the Irish EEZ. Then, as I mentioned before, the resolution is a very big factor to take into account here as well. Ideally, we would look at a finer scale, but have information on the whole Irish fleet. The type of data that was used here, in this case, the fishing effort, might also not be the most suitable one to here assess the risk of entanglement. When we look at fishing hours, we know how much the vessel has been fishing, but we don't actually know how much spots are in the water or how much rope is in the water, which is what we're interested in. Also, in this case, the landing records that were used to calculate the number of pots appeared to be an underestimation of the actual number of pots. When the Marine Institute looked at previous questionnaires that they had done with fishermen, we appeared to have a much lower number that one was actually obtained from questionnaires. So that's something to also consider and to take into account. Then for the entanglements themselves, as we know, there is a very big underreporting, and specifically also probably for the entanglement-related strandings. And at this point, we also don't have any information of the live entanglements that happen at sea, or that floating animals that end up in gear and that never end up on the shore. So our recommendations would be to, in the future, use other methods to assess the pot fishing effort, such as recording of surface buoys, but also using questionnaires with fishermen, because this has been very useful in other studies that have been done in Scotland, or also use novel methods such as aerial surveys or using unmanned aerial vehicles or satellite imagery. Also, we need a better capture of the number of strandings and entanglement-related strandings. The Irish Whale and Dolphin Group has been working on this with better trainings, better quality of pictures, but obviously other resources and post-mortem evaluations would help here as well. And then additionally, insights into incidences that happen at sea would give a lot of information there as well. And this could be done through also questionnaires with the fishermen, or having a reporting scheme that would be used by the fishermen. So to conclude, this study was able to identify areas in the Irish EEZ where humpback whales, minke whales, and fin whales have a higher risk of co-occurrence with pot fisheries and therefore a higher risk of entanglement. We believe that these areas that were identified in this study could be used in the future as priority areas as to where to concentrate future efforts. At this point, we were unable to really assess the impact that entanglement is having on the whale populations, but we were able to identify gaps and constraints in the study so that in the future, so that recommendations could be made so that in the future, the studies could be more focused on using that type of data and to be able to identify whether or not pot fisheries are having an impact on whale populations in Ireland. Thank you for your attention.
Okay, thank you for your talk.
Thank you.
Yeah, it's, maybe the title is not so accurate. You are focused on entanglement, and the real conclusion is that no entanglement can occur, or are not seen.
That we don't have enough data at this point to be able to say, because we have, over the period of the 34 years that we looked at, we saw 9 cases of entanglement. But like I said, we don't have, or we're lacking a lot of data on life entanglements and stuff as well. So for example, another study focused in Scottish waters, pretty much 60 or 65% of assessing whether or not entanglement was an issue or not came from data from fishermen that reported entanglements at sea, but not from the actual strandings. So that's, obviously, yes, there is a big lack of evidence to be able to say whether or not this is a problem.
The establishment is perfect. I mean,
no.
The distribution of this method, but the distribution of the species, because all the species, they are more or less in the same place. So I was thinking, come on, they were didn't increase the number of entanglements or significant number of entanglements in order to change the fisheries politics or the restriction of fishing areas in Ireland. Yeah, we need that. That's what basically the study also. It's the first step towards being able to do more and to be able to go afterwards to the fisheries saying this is a big problem.
But yes, it's definitely a first step because in Scotland they're already at the point where they're going to do like have mitigation actions being taken with the fishing, changing fishing gear and stuff. But in Ireland, we're far behind there. And this is obviously only the first step. Yeah, for sure.
Is spot fishing in shallow waters or in deep water?
No, they're also in very shallow waters. Yeah, because when we were out on the boat and stuff, we were only like 50 meters away from the coast, and we would see the pot fishing.
Big whales, they come.
We see them there as well. Yeah, in Ireland. Yeah, because even we did also not specifically related to the thesis, but I did on other like participating in land watches and stuff. And there is a big area where we spot whales just from the coast. Yeah, yeah, yeah.
Okay, good luck.
Yeah, I know it's a very big yeah. Still a lot of work and yeah, and not easy to.
You're thinking some kind of
Sorry?
recording of the areas. Okay, I don't know. Record. Yeah.
Yeah.
Well, good luck in it.
Thanks.
I'm a listener, but I am very curious. Is your risk anything more than just a co-occurrence, or do you assume? I mean, how do you get from the co-occurrence
to risk
a
of
probability of?
No. You're totally right. No, at this point it was only this. Yes, a lot of other studies have done the same thing, where you basically also do an overlap and do the risk of entanglement measure or measure the risk of entanglement. And they also had the strandings data to back up this information, obviously, to be able to say we have these risk areas that have been identified, and we also see that there is more like strandings that are related to entanglements that are happening here. But because we're lacking so much data there, we can't back that up. Yeah. So yes, that's why we're also talking about risk of co-occurrence. In these other studies in Ireland, they made this link to say it's a risk of entanglement measure, but it's more risk of co-occurrence, obviously, because you can't at this point not yet link it to entanglement. Yeah.
Yeah.
We got the basic, like the first, yes, yeah,
and
for sure.
How did you? So you had sightings also. How did you integrate the sightings data
into the risk assessment,
yes. or did the sightings data?
So we first like
all what we meant.
Yeah, so we used the sightings data, which were over 24,000 sightings, both effort and non-effort related, which was, well, a very big part of the data as well, to use opportunistic data in this case. And then, yes, we mapped them out on the maps of Ireland, and then basically, yeah, overlay the grid to be able to then count per grid size how many whales were present per grid, yeah. And then overlap this, yeah, with the fisheries effort, yeah.
Two questions.
Yes.
About the fisheries.
Yes, of course.
One was you said you. So what about recreational fishing? You said you haven't covered all of it, but you must have. There must be recreational fishing.
Yes, definitely.
So do they need a license to qualify those?
No, we don't have any information on those. So these are only the commercial fishermen. And so that's why what they did in Scotland with using recording of surface buoys, then you would include the smaller fisheries or or recreational fishermen. Yeah, that's why that's one of the big recommendations, and also basically what my promoter wants to go to afterwards as well. He wants to make this a bigger project, ideally a PhD of several years, to be able to also quantify all the broad fishing of broad fisheries, but both of bigger vessels, of the small vessels, of the recreational fisheries. And obviously, if you would do this recording of the surface buoys, or with satellite imagery, with just doing surveys, you would get all the information at once. You would.
Do you have licenses? Do you need a license? I would think that, yeah. What about the number of licenses?
I honestly would have to look into that for the recreational fishermen, because I don't think, because I was in Ireland a little bit as well and talked a little bit with fishermen, and they would just go out and put their boats. But I don't honestly. I would have to look into that if they need a license for that, because I know that the people that I've met were just going out there and putting them out there. So I'm not sure. I would have to yeah. I have like I didn't come across any other studies that would use something like that. So I'm not sure. No, honestly, I would have to go back into that.
Honestly. But and the final question about that is, are there really international fishermen
Yeah,
in
yeah.
Irish waters?
Yes. Yeah, Belgian, German. Yeah. Yes. Yeah. And obviously we weren't able to include those because they don't have to report this to the Irish Naval Service and stuff like that for wherever they're fishing. And we also expect them to be bigger boats that have a lot of bots that are deployed. So it would be interesting, obviously, to also be able to include those in the analysis. Yeah.
So it's very interesting. It's kind of even
Thank you.
And I wonder because you say that also I don't know if you say the percent that you have what's next to the coast. So I wonder if the entanglement or the stranded is the result of the entanglement or is the other way around. They get entangled because they are too close to the coast
To the shore.
for any reason.
Right. Well, like based on other studies that we've also like compared with like just normal abundance data on the whales in Ireland, they come really close to the coast. Yeah, they are inside like sometimes the 5 nautical mile area. So yeah, they come really close to these areas, which has been shown for over years already. So it's obviously we're putting the pots over there, so it's our fault that they're getting entangled. But yeah, they have been present there for a very long time. What's been happening though as well is that they're migrating more up north as well with climate change and stuff. So that's also something to look at and to look at the variation. And over the years, maybe some areas are identified now as risk areas, but in the future, if we for example, would put all our pots more up north, that could be a potential other risk areas in the future if the whales migrate up north.
So that will happen.
We don't have that information, no. And in Scotland, that's something that they looked at also very closely. And like I said, like around 65-70% of the data where they were able to say, they get entangled and to back up the information about here, these are the risk areas, came from that information, from life entanglements that fishermen would report, or for even having an institute that actually, whenever a life entanglement is reported and they can do something to help the whale, they would that's also recorded, and that's also something that they would use to identify.
Because once they entangle, they are able.
Depends. The humpback whale is a stronger whale that can more easily get free, but the minke whale is, for example, one that gets in, like if it gets entangled, the chances of Oh.
So you've been speaking about entanglement. I know you have where you have sort of stranded, but do you know which part of the body or how they?
Yeah, so well, there is a little bit of research on that as well. It depends a little bit on the species, mostly. For example, the minke whales, they would mostly be entangled around the mouth and the head, while the humpbacks would be mostly around the fluke and the tail. But why or how, that's uncertain. They also did a study or like looking at which part of the ropes would get entangled around the whale, if it was more the vertical rope or the ground lines. And there it appears to vary as well. Like in the northern right whales would mostly be the vertical rope going up, while for humpbacks and minkies, They had studies in Korea, but also in Scotland, where pretty much 80% ended up entangled in the ground line, which is not really what you would be expecting because the vertical one is like the most obvious one to get entangled to. But yeah, these ground lines are also very important lines to, that lead to the entanglement of the whales. Yeah. Thank you.
Very warm welcome to everyone, and thank you very much attending my master's thesis presentation. I'm Sawandi Alvis, presenting you investigating gastrointestinal parasite infections of common dolphins in relation to a range of dolphin health parameters. In the wild, the health of cetaceans is affected by a multitude of anthropogenic and natural stressors. Among anthropogenic stressors, the fisheries in bycatch in fisheries, such as in figure A here, and vessel strikes, figure B here, and also shipping and wildlife ecotourism can elicit stress responses in cetaceans. And if I talk about the natural stresses, predator attacks, and the competition for the same prey species, in the figure A, we can see that the gannets and dolphins are competing for the same prey resources. And along with environmental changes and also parasitism can all pose challenges to the health of cetaceans. And parasitism is the association between two different organisms wherein one benefits at the expense of the other. And parasites are ubiquitous. Parasites are ubiquitous. They play an integral part in the ecosystem and affect the ecosystem functioning and also enhance the biodiversity. In cetaceans, many microparasites have been observed. They are virus, bacteria, protozoa, and fungi, and also macroparasites also have been reported. They are helminths and arthropods. Parasites in common dolphins and also in general cetaceans can Parasites in cetacean can go. Can I have a minute? Parasites in cetacean can transfer trophically via the food web. If I talk about the effects of parasites in marine mammals, up to a certain threshold, parasites can impose localized reactions. For example, in this picture, the parasite structures are surrounded by the inflammatory cell infiltrations. And more severe forms can occur, such as granulomas, hemorrhages, and ulcers. And in this picture, we can see anisakis infections by common dolphins, and it is a severe form of ulceration. And sometimes death of the host can occur. For example, cerebral nacitremosis caused by nasitrema, which is a trematode, and this trematode can cause inflammation and death in brain tissue. And sometimes the influence of parasite infections can go beyond the individual level and affect the host population. In this paper, the authors try to highlight that the lungworm infections can transfer from mother dolphin to the baby dolphin in the womb itself, and in these cases, neonatal mortality is expected. So coming back to my species, common dolphin, they show global distribution, and it is one of the most abundant species in the Northeast Atlantic waters. They show an year-round presence, and it is the one of most abundantly recorded stranded species in the Irish coastline. And between 2017 and 2019, 78 common dolphin carcasses were collected as a part of the vertebrate necroscopy scheme. So these were the samples that I used for my research project. So in order to comprehend the pressures faced by marine mammals, it is crucial to gain a deeper understanding of their parasite loads and resulting impacts. So addressing this, our study aimed to investigate stomach parasite burden of common dolphins and examine the relationship between the parasite abundance and health-related variables. And additionally, we conducted a histological analysis of gastric ulcers. So, first, we dissected the stomach of the common dolphins, and then we recovered all the parasites from all the stomach compartments, and we preserved them in ethanol. And subsampling and subsequent estimation of parasites were carried out using the equation, which is wet weight of the parasites in the sample divided by the wet weight of the parasite in each subsample multiplied by the number of parasites in each subsample. So before I move into the results section of the first section, I would like to introduce some terminology, which is the mean abundance. It is the total number of a particular parasite divided by the total number of hosts. So in this example, it is 3 divided by 3, which is 1. And the prevalence is the number of infected hosts divided by the total number of hosts. So in this example, 2 divided by 3, or else we can say it's 67%. And the mean intensity is the total number of a particular parasite divided by the number of infected hosts. So in this example, it is 3 divided by 2, or we can say it's 1.5. So moving on to the result section, we identified the parasites as nematodes belonging to the Anisakis genus. We identified them morphologically. We could not go into the species level. And the figure shows adult Anisakis specimens, and I will go through in detail if anybody is interested really. And 456,422 Anisakis specimens were estimated, and the mean abundance was 5,850 worms, and the prevalence was 89%, and the mean intensity was 6,610 worms, and the range varied between 2 and 71,115 worms. So moving on to the second section, which is examining the relationship between the parasite abundance and health-related variables. Generalized linear model framework with negative binomial error structure was used to analyze the potential predictors of parasite abundance, and all of these 11 predictors are of common dolphins. And if I move on to discussing more about these variables, nutrition status had 4 divisions, which is very poor, poor, moderate, good. And the cause of death also had 4 divisions, which is trauma, infectious diseases, starvation, and others. And the decomposition state of common dolphin again had 4 divisions, which is extremely fresh, slight, moderate, and advanced decomposition. And the maturity of dolphin we divided into 3, which is adult, juveniles, and calves. And we removed the weight of dolphin, length of dolphin, and maturity of dolphin following the data exploration. And first, a full model was built, and then reduced models were built, sequentially removing the variables which had no significant effect, which is also here, and which were less biologically important, which is decomposition state of common dolphin, resulting in a final model. So the results show that year of stranding of the common dolphin, age of common dolphin, cause of death of common dolphin, nutrition status of common dolphin were significantly associated with parasite abundance. So the parasite abundance differed significantly between 2019 and 2017 only. So the observed temporal variation in the parasite abundance could be due to the prey availability to common dolphins, and also we need to investigate further the prevalence of Anisakis in the prey species of common dolphins. And here, if you look at the graph, x-axis is the age in years, and the y-axis is the parasite abundance, and we can see a moderate positive correlation between the variables. So it suggests that older dolphins tend to harbor a higher number of parasites when we compare it to the younger individuals. The reason to this could be the mature adults need more energy so that they may encounter the prey species that has anisakis inside them. And the parasite abundance differs significantly between poor and very poor nutrition status of common dolphin, poor versus moderate, and poor versus good. When we look at the box plot, x-axis is the nutrition status and y-axis is the parasite abundance, and we can see that the common dolphins with poor nutrition status had the highest number of parasite abundance. And in general also, common dolphins with very poor and poor nutrition status had higher abundance of parasites when compared to the better nutrition status of common dolphins, which is moderate and good. So that we can say that common dolphins having better nutrition conditions were resistant to the parasite increase in parasite abundance. And the parasite abundance differed significantly between infectious diseases versus trauma, of cause of death, and infectious diseases versus starvation, others versus trauma, and others versus starvation. And if we look at the box plots, again we can see that the infectious diseases was the leading cause of death among the dolphins. And it is not clear to what extent the anisakis have played a role in death of trauma, starvation, and others. There was no significant relationship between parasite abundance and sex, and similar results have been reported for striped dolphins, bottlenose dolphins, beluga whales, and franciscana. And this similarity could be due to the similarity in diets of common males and female common dolphins. And moving on to the third section, we wanted to identify the histological characteristics of the affected area and the healthy tissue. So 2 tissue samples were taken using a core of 5 mm width, and they were processed using a they were processed in alcohol and embedded in paraffin and cut using the microtome at 4 micrometer thickness, and they were stained in hematoxylin and using. And finally, slides were examined in the light microscope at 100, 200, and 400 magnification. And if we look at the healthy and the severe gastric ulceration tissues, we can see that the severe gastric ulceration tissue had abundant inflammatory cell infiltration along with parasite remnants. And a suspected Anisakis eggs were also found in the exudate of the ulcers. So implications, the results provide insights into marine wildlife management, conservation, and monitoring programs. For example, if we incorporate parasite monitoring into the existing health evaluation of cetaceans, we can identify the trends of the health status of cetaceans, including common dolphin. And another thing is, if there is a possibility that we can use parasite abundance as an indicator of the body condition of the hosts. And future directions, investigating the diet shift of common dolphins due to prey population dynamics, climate change, and anthropogenic impacts and their potential effects on parasite loads are important areas of future research. And it will be necessary to assess effects of multiple stresses and their accumulative effects on common dolphins along with parasitism. In conclusion, the results highlight the importance of considering parasite infections along with other health parameters when evaluating the health status of marine mammal populations. It's my acknowledgement. References. Thank you.
Thank you very much. Even though it's on time. So how much of it were you involved in? To actually sum up the dissection?
Actually, no. I did not involve in this project. I mean, like involved in the cutting of the stomachs. My PhD supervisor did that for me, and they had collected all the parasite specimens, and I did other work. I mean, the analysis and everything.
Okay.
Yes.
And did you do histology?
Yeah, I did the histology part as well.
Which is also not. It can also be quite messy.
Yes, it is stressful. Yes.
I'm curious about the chicken. I find it a chicken and egg. You know, there's a certain.
Yeah.
Do dolphins who are sick get parasitized, or do parasites make them sick?
Yeah, that's why.
Can you talk about that a little?
Up to a certain threshold, there can be only local reactions, and there won't be systemic reactions. But if a threshold is exceeded, there can be, as I explained, there could be granulomas, hemorrhages, and ulceration. And in extreme cases, there could be death of the host. And I think it depends on the parasite species as well, and also the host species and the burden of the parasites inside the host.
Okay. But what about the other one? If it's sick, dolphins are more likely to parasitize.
Yes. If the nutritional status and all their other conditions are bad, and if the individual is very sick, I could say very weak, the infestation of the parasite could weaken them further and finally could lead to death as well.
And I guess the one missing piece of information in this is how do dolphins get anisakis?
Anisakis, yeah. I explained that they could transfer trophically, I mean, from the food web.
From which species?
There are specific fish species that the common dolphins prefer. So from those prey species, and common dolphins can get anisakis. I mean, if the fish species is infected with anisakis, they're going to get anisakis infestations.
What species are they?
Herrings, especially.
Anything else?
You mean the
other fish? I mean, herring's the famous one.
Yeah, herrings. I could say sardines, definitely. Herrings, sardines are the species. Yeah.
Okay, nice work.
Thank you.
I like this very much. I'm a biologist.
Okay.
I do histology a lot. I'm sure the last 1 year and a half we are running stranded mutation in Embrc in this country. So we have been doing this year many necropsies.
Okay, I love necropsies.
And I have seen more or less the same size anisakis that we have seen in the gastrointestinal tract, but not only there, but also in the lungs.
Yeah, okay.
50 centimeters anisakis there. So one thing is that I was thinking in the sex, maybe that we made it with the prevalence was, for me it's very important not only the prevalence, but the intensity, as you say.
Yeah.
It's not because the prevalence can measure only one single parasite in one single animal.
Yes.
It's not so important. Well, it's important, but it's not the information that you are looking. So So it's good to see that the intensity of infestation have been checked. Also, it's quite clear that the spec that old animals they've got more parasites. Also, juveniles they are not so exposed. But the cycle of the anisakis is quite complex.
Yeah.
It's very, very complex.
Yes.
Including mollusks, including species. We did some work some years ago, and we checked that 80% of the fishes species, at least in the Bay of Biscay, they are infested by anisakis. And the target host is the mammals.
Yeah.
So the dolphins are the target.
Yeah.
So in some cases, there are some doubts saying, okay, they can stand the massive presence of parasites.
Mm-hmm.
So one of my questions is, okay, which are the parameters you say the title that you were comparing with the infestation and seeing the in relation with health parameters?
Yeah.
Which health parameters did you check?
These are all the health parameters, like health parameters in the sense they are related to the health of the common dolphins, especially the nutrition status. Cause of death also can be a health parameter, why they died, and the age is also, and sex and the presence of ulcers. We can get an idea why ulcers are present in some stomach samples, why not in some, and weight, length, and maturity.
Do you think that these are health parameters?
I'm sorry?
For me, health parameter could be the immune system.
Oh,
the
yeah.
expression of the immune system. These are different clusters of information that can help you to understand the prevalence.
Okay.
Because don't forget that you are analyzing animals that are dead. You don't know, as the previous member of jury asked you don't know if the animals died because of parasites, because they were weak, or because it's nothing to do with interaction with the fisheries.
Okay.
Because they can be very healthy.
Yeah.
And they crash with the hull of the boat, and they die and they strand.
Yeah.
So that could be something that you could analyze whole matrix of your data just to discriminate different sources of information.
Yeah, and the other thing is like we have used the stranded dolphins, so it is kind of biased because stranding could be due to the injuries sustained and could be sick individuals most probably. So it's kind of biased, but I must say that it does not like represent the whole population of common dolphins, but we can get an idea.
Also, you discard the protein status of the animal. That's important.
Sorry?
The status of the dead body.
Okay.
The composition state.
Yeah.
That's important because if the animal has been dead for a long time, floating or being moved by currents or whatever it is, they can increase the amount of parasites inside. And if you discard when the animals they are stranded on the beach, you discard the ones that are frozen.
Okay.
Maybe you are introducing a bias in the analysis.
But actually, like we are exploring the stomach dolphin, stomach parasites. So decomposition status is not that biologically important because.
Why not?
Because, as you said, that they could be floating, but how could anisakis go inside?
Can be eaten by different predators. They can open the cavity, the stomach cavity, and parasites can come in.
Okay. Yeah.
I'm giving you ideas.
Yeah, I understand. Exactly.
Your further research. Okay? And also the last one is you should improve a little bit the quality of your histological section. Okay. There are some of the, according to the micrographs, some of the sections where the micrographs, they've got the light, not very focused in the middle, got some black parts in the you see over the scale bar.
Yeah. Okay.
The light is not very focused. Widespread section. So could be a problem to increase the resolution and decrease the contrast.
Okay.
And maybe sometimes it's crucial to obtain high quality samples.
Okay.
Because you have to differentiate between artifacts and histopathological alterations.
Okay.
Sometimes you can fix them if the processing is not very good. But my advice is to do the proper protocol to obtain a good fixation. In this case, I think the fixation is longer than what it could be the best.
Okay. Yeah, and the other reason could be these samples were obtained in 2017, 2019. So they were in the freezer for a long time as well, and the thawing and everything.
So they were frozen, and then you do the fixation.
They were frozen and they were thawed, and then we took out.
The recommendation is do as fast as you can.
Exactly. Yeah.
Do the fixation.
Yeah.
Maybe you cannot do because you are on Sunday or what Saturday or night, but the next couple of days to start and do the process and go up to the paraffin blocks, and then you can maintain there for years.
Okay.
Yeah, that's some
Thank you for the insight.
Thank you very much.
Thank you.
I'm going to be very. Thank you very much for your presentation. It's kind of exciting. I have a mine is just curiosity because I saw one of the graphs you show us. I'm not sure if I understood the graphs. Is when you show the arguments of anisakis during time between different and I think that maybe the abundance goes down. I mean, it's higher in 2017 and
yeah, yes.
And as far as I know, but just knowledge, you know, it's gonna happen that the anisakis is in the person's
yeah.
And showed me that the fact that instead of increasing the presence of the abundance of anisakis is related with the cycle of anisakis is something.
Yeah, exactly. That I explained. I mean, like we can clearly see there is a temporal variation here. 2017 is being higher and 2019 lower. To investigate that further, we need a lot of data about the prevalence of anisakis in prey species of common dolphins. So we can get those data and we can give an explanation for this.
Should be a little bit maybe hoping they keep more stable.
Yeah.
Maybe not.
Thank you so much.
So this is the end of the session. The end day, long day, and I would like first to thanks again after the students' discussion of the project, which I am national and on West, and also was sending and studying areas and times of the world that is very and finally, the one study version I see concept along other parameters and so this thanks again.